Okay, well, as we kick off, this is the 28th session of the Scientific Review Board. I just want to note that Anna has sent apologies. Unfortunately, she's quite unwell. She was going to join us online remotely from Finland, but she's just called in that for personal reasons she won't be able to join us online today. Hopefully, she'll be well enough to connect tomorrow. So we look forward to that. With that said, could we just bring up the agenda on the screen, please? Wonderful, thank you. So the first thing to notice is that we do not have a chairperson. Sean Cox retired from the SRB last year, and we've been waiting to get a replacement on board before we were to actually elect a replacement chairperson. With Anna having been away, not available, she has sent a nomination, however, for Olaf. So if there's a second for Olaf, there is. And are you willing to accept? Congratulations, Chair. Okay, well, I'm going to pass to you then, and then we can do an introduction of Mike as well. Pass to you, Chair.
Sounds good. So we will move on to the adoption of the agenda and arrangements for the session. Any changes to the agenda since this was posted? That's all. Okay. Shall we do introductions now?
Yeah.
I can start us off. So I'm Olaf Jensen. I'm a professor at the University of Wisconsin-Madison. I've been an SRB member since 2020.
Yeah. Ian Stewart. I do the stock assessment, work closely with Alan and Ray. It's part of the quantitative team here. And I started with the commission in 2012.
Alan Hicks in the quantitative sciences branch on MSc in urban strategy policy, and been here 10 years now.
I'm Andy Jusanowicz. I'm a research biologist in the biological and ecosystem sciences branch, focusing on genetics and genomics.
Morning, I'm Giuseppe Plemes. I'm the manager of the biological and ecosystem science branch. I've been in the commission since 2016.
I'm Claude Dykstra, research biologist within the biological ecosystems branch. I work on a variety of projects from discard mortality to kind of fish movement and catch protection.
I'm Colin Jones. I'm a research biologist in the biological and ecosystem sciences branch, and I'm primarily focusing on reproductive biology at Pacific Health.
Pasha Nichak, I'm fisheries regulations and data services branch manager, and in this role, I coordinate development Pacific Health regulations annually and compilation of data that goes into various IPHC data. And I'm here since 2019.
Mike Wilberg from the University of Maryland Center for Environmental Science. My background's in population dynamics, stock assessment, management strategy, evaluation. Do work on all sorts of different species, from invertebrates to fish to marine and terrestrial mammals to birds. I like to model whatever I get my hands on. So I'm excited to be here. Thank you for inviting me.
Ray Webster, nutrition statistician. I work a lot with this data, science. I bring the stuff on analytical needs. Almost 20 years coming in October. It makes me the second longest staff member.
Do you have a couple of staff members? I think we're just hitting 28 years.
John's over 30.
No, I think John is
38 years.
Yeah, Jones over 38. Yeah.
Just a decade. Okay, and Dave Wilson. Since 2016. Yeah, so we have two observers online, and these are our science advisors from Canada and the US. Anne-Marie, do you want to introduce yourself?
Good morning. My name is Anne-Marie Huang. I work for Fisheries and Oceans Canada. I am a salmon biologist moonlighting as a halibut science advisor.
Thank you very much. And Pete?
Yeah, thanks, Dave. Good morning, everyone. My name is Pete Holson. I work at the Alaska Fisheries Science Center for NOAA Fisheries, and I'm a stock assessment scientist for groundfish. And yeah, I'm the advisor for the U.S. delegation.
Thank you very much. And just another quick note as well that we have Dr. Joanna Mills Fleming from Dalhousie University joining the SRB. She was contemplating coming and joining us for this meeting, but due to other commitments, she's going to start her term for the SRB in September. So you'll get to know her as well. If you are interested, bios of both Michael and Joanna are up on the website. And later tomorrow morning, I think we have an offline session, and maybe Michael would be useful if you were to give sort of a little bit more of a detailed background as well, so sort of couch the discussions around your experience and expertise. Okay. Future.
Great, I think we're on the IPHC process then.
Fantastic. So in terms of IPHC process, and this is particularly for Michael's benefit, we have 2 SRB meetings a year. This earlier meeting is really designed for the board members to sort of critique what's being developed and analyzed by the Secretariat staff, and to provide sort of a recalibration, a redirection, or just general input. So over the course of the next 2.5 days, you'll have an opportunity to provide direct feedback on the papers that are on the website, but then also the offline session that I was talking about. And that's something we've done informally in the past, but now we're locking it into the schedule because we think it'd be particularly useful for SRB members to engage with some of the staff on various projects that we're undertaking offline. So it gets a little bit too formal online. So it's unfortunate that Anna can't be with us. But primary purpose of this meeting is just to provide that redirection. Generally, we don't develop specific recommendations for the commission at this meeting. It's really about requests for redirection suggestions. And then that meeting in September is where the SRB provides that formal critique or peer review of the work that's been undertaken, and then provides clear recommendations to the commission, whether you agree, whether you think we should take a more structured adjustment to what we're doing, and if there are any specific recommendations that we're seeking peer review on to go to the commission. So this one's a little bit more casual, which is good. That's it for annual workflow. The chair will present your report from the September meeting at the annual meeting, which is the last week in January next year, so the 22nd of January. So the first day of the 25th of January, Olaf will present your core findings or recommendations to the commission. We do have an interim meeting, which this year is on the 2nd of December, I think it is. And that's also an opportunity if you are available, and that's purely on feel that there's something that you really want to push and notify the commission of. That's also an opportunity. Otherwise, we just provide the SRB reports for information. In addition to that, in September, we have a work meeting. This year, it's going to be here at the commission headquarters. And again, it's that sort of informal opportunity. If the SRB feels that you'd like to raise something to bring to the commission's attention, it's also an opportunity. But if not, we just provide them with a report of this meeting for information purposes. That's it on the workflow. Typically, we just drop in the core terms of reference for the SRB, the mandate, into the report itself. It's the second paragraph, just to refresh our minds of what the SRB's core functions are, but then also anyone who picks up the report in the future or any time, so they are aware of what the core is. Could we have the next paper up on the screen, please? Paper 3, which is update on the actions arising. Again, I'll go through a little bit more detail, Michael, so it's for Michael's benefit. This is a standard paper that we have for all of our subsidiary body meetings and the commission meetings themselves. Just a specific, very clear update on the requests, recommendations, or any other actions from the previous meeting of this. So the design is pretty straightforward. common for most RFMOs in the sense that we really do try and focus the requests and recommendations to have a very specific action or deliverable. Who's going to undertake that action, responsibility for it, and then hopefully a desired time for if additional funding is required as well. So provided at Appendix A is a table of all of the recommendations and requests for action that arose from the previous meeting. And so the way it is designed is you have the meeting, the recommendation number, what that specific text was, and then we provide a very brief update so that we're not getting too extensive in this particular document. Most of the time, what you'll see is that there is a reference to a document that's before the body itself. And that's where we provide either the progress report or if there's a specific delivery. So in some places you'll see it linked. In other places we didn't get around to I usually don't step through these in detail because as I said, each of them are going to be addressed in a particular agenda item. One thing to be mindful of is that as we progress through the meeting, unless the SRB revisits and wants these recommendations or requests reiterated, if they haven't been completed, they usually just get dropped. So if it's a case of, no, we see that this is in progress, we see the work that's been undertaken, we want you to be redirected this way, and you put it in as a new request or recommendation. If you're content with what's being progressed and the work plan, generally just leave it. That's your discretion.
Dave, can you just remind us, request versus recommendation?
Yeah. So we have, and I might drop that to you later this afternoon when I provide you the report framework for drafting for this meeting. We have a number of formal language that we like to use in the reports. Requests for action are those sort of informal requests that may or may not be possible, and you're just trying to direct the secretariat in a particular way, or you have a request for a particular analysis to be done before the September meeting. The recommendation is a formal action that you want the commission to be aware of, even if it's to us. It's a case of we recommend that the the secretariat undertake X, Y, Z. We want to make sure the commission knows that this is being requested. So it becomes more of a formal action. And then we have some just sort of general terms that we try and describe for clarity and consistency. But those are the two primary terms: request, informal action; recommendation, formal, in the sense that the commission is going to go to the commission as well for awareness.
We also use noted just to bring attention to a particular item. Often that precedes the recommendation.
Awesome. Are there any questions on what's in this particular document? See that there's only one. So still 5 there that are in progress.
A lot of them are mine.
So assuming that you've had a chance to review it, any questions or comments from any of the team as well? Additional updates, so you just wait. No? Not seeing any? Keep my eye online as well. Okay, well, thank you very much. That's it for that paper. If we could have paper 4 up, please. So similarly, or in a similar format, we also provide the outcomes of the most recent annual meeting of the Commission. Again, this is a reference document. We don't typically go through this in great detail. So there are meetings of the Commission where there are very specific directives coming from the Commission. At this particular meeting, as you can see in Appendix A, there really wasn't. Most of the work was done for the Harvest Strategy Policy, for example, at the interim meeting. And other components were really just the Commission taking note of what was going on. Some years in the past, we've had very heavy actions by the Commission in relation to the work of the SRB. But I'm going to say, because things are progressing very positively, smoothly, really just be noting what's been going on and haven't provided any particular redirection either to the Secretariat or to the SRB. The caveat to that, or the only one rather, is the FIS design. Sounds like you were already discussing that on your walk in this morning, where there's been a lot of activity by the Commission in terms of deciding what's not only scientifically, I shouldn't say only, what the scientifically robust design is, what can be implemented from an operational perspective, finance, that type of thing. So they've been putting a lot of time into reviewing and making ad hoc adjustments, but I think we're at this point now, and Ray's going to talk to you a lot about this. We're at that point where now we feel we can implement the base block design. So we're still able to headed back to you.
It's a fun multi-criteria optimization problem.
So that's this paper in a nutshell. It's really just there as a reference. It will be provided to the SRB in fall as well, just again as a reference document, just to provide you that ease of access to those for decisions or information sessions that went on.
Just a quick question on it, and if I realize I'm learning stuff as I'm going here, so if this is better off later on, that's fine too. But how does the commission interact with the SRB? Does the commission come up with things that they say we'd really like the SRB to look at, and that kind of ends up being a task between the Secretary and SRB to do, or is it mostly SRB identifying things the commission should be aware of?
It's been a bit of both. It's fluctuated, I'm going to say, over the last decade. At times, the Commission has requested a very specific task, such as peer review of the stock assessment or the MSE work. But generally, they're just monitoring what's going on and taking feedback from the SRB in interpreting the recommendations or the analyses that we're providing. So, for example, if EM provides a stock assessment, they may go and read what were some of the clarifications or requests for modifications or inputs to the stock assessment throughout the course of the year. So I would say at the moment, it's very much just a monitoring side of things from the Commission. Does anybody off the top of your head recall any very recent directives? I don't think there has been. I think often the last couple of years, it's been papers like this, where it's just the Commission noting the information coming out of the SRB in relation to the scientific work that we're doing. There was probably a couple with the harvest strategy policy analysis, but you're going to cover those.
Yeah, but I think a lot of the interchange between the commission and the SRB occurs when the chair is presenting SRB report as well.
And the FIS design, as we've gone through the design process and that's evolved, they've had specific questions about how that process works, what's the base for our design process. They've added specifically for the SRB to review components of that as we've gone along. But I think that at least the process side of that is kind of set.
But we don't get specific terms of reference like we do at council.
Okay. Well, that's it for this paper. I could just bring the agenda back up again. Please. So typically at this point, we turn to our two science advisors to provide some of that additional clarification that may have been coming from their commissions since the annual meeting itself. And so if we will just pass to Anne-Marie, if you would like to, just sort of run us through any additional updates or questions, comments from commissions. Anne-Marie.
Thanks, Dave. Nothing specifically from the commissioners. I've been away for a little while, so I haven't had a chance to really catch up with them. But maybe from the Canadian contingent overall, two main themes that I've been hearing about that does overlap with some of the things that I've heard from the commissioners over the past little while as well. One is one that you guys were just talking about in terms of the FIS footprint and sort of questions around the space-time model and FIS interactions, the way that it's sort of been asked me is like, what's the breaking point? When do all of the holes in the FISP footprint end up affecting the space-time outputs? It's been phrased to me as a breaking point, but I think in my head it translates into, yes, the outputs are wrong but useful. When does it become wrong but not useful? And also, I think what happens is that it just sort of goes towards long-term average. So when does that happen? Do we get an idea as to that? And then what are the implications for what gets put in front of the commissioners? The other theme that's popped up is the concept of depleted. So I know the commissioners were wondering about, where else has it been used? How has it been used? What were the outcomes of implementing the depleted concept? Last week from MSAB, I think something that came up, it was around the management procedures that operate both in relative spawning biomass space as well as absolute spawning biomass space. Everyone's sort of trying to wrap their heads around how this functions, how this works. And from a management biologist perspective, it's like super fun to think about. But then it's like there's a little voice in the back of my head going, do we really want to go down this route? Is there a simpler way? Is this going to, does this necessarily are we unnecessarily complicating things just because we can? And so I think that's something that's sort of popping up a little more recently. Thank you. And Olaf, Dave, I'll draft up a couple of points for you to consider to put into the notes so you don't have to scramble and try to interpret what I'm saying.
Great. Thanks, Anne-Marie.
Steve. Any questions for Anne-Marie before we go to Pete? If not, Pete, go ahead.
Great. Yeah, thanks, Dave. So from the U.S. Commissioner side, there aren't any specific questions at this time, but that is to say that they are all very interested to hear the discussion and the analysis and investigation as it proceeds on what depletion is and how we're thinking about it, and specifically concerning just the current status of the stock and how that relates to depletion. So no specific questions at this time, but they will be keenly interested to hear what the SRB has to discuss on this topic at this meeting and in future meetings. Thanks, Dave.
Awesome. Thank you very much. Any questions for Pete? If not, I'll just also note that we have Courtney Young from DFO in Ottawa online. Courtney, you're very welcome to introduce yourself, or at any point in time, just raise your hand if you'd like to ask any questions.
Thank you.
No worries. It's easy. Okay. That's it for observer updates. So we could go to the next agenda item. Agenda item 4. This is Big 55. Just have paper 5 up on the screen, please. Okay. So this has been ongoing for a while now in terms of trying to finalize and update the integrated program of research and monitoring. This plan has been designed to replace the previous 5-year program of research and monitoring at the Secretariat. We've been going through quite a number of iterations with the commissioners and with the board itself over the last number of years to get it into a format that we felt we could adopt it, finalize it, and replace the previous. So as of earlier this year, following the commission meeting where they reviewed the various recommendations that arose from the previous SRB meeting, I think those recommendations came in the June meeting of the SRB. We made a number of improvements to the plan itself. I don't intend on stepping through all of those. It's really at this point now going to become a reference document for you. If you haven't already had a chance, Michael, to go through it in detail, I would just recommend that just read through the plan, give you a good idea of how the Secretariat is structured, our core functions, and what we're trying to implement, at least in the near term. And then there are some sort of broader, longer term activities that we've outlined based on mission goals and objectives. It is going to remain as a working document. Each SRB meeting, particularly the September meeting, we're going to look to see if there are additional direction or ideas the SRB would like to see the Commission consider for implementation. And once the Commission has had an opportunity to consider those recommendations, they may well indicate that, yes, you should replace this activity with another research stream. That is also in line with our budgeting process, where we take our 3-year budget proposal to the Commission, to the Finance and Administration Committee in January. It then goes to the Commission, and if there are any other research streams that we need to implement, they'll be incorporated into that proposed budget. And that's where it's approved for the subsequent fiscal year starting October 1. As I said, I don't intend on going through the plan itself. It's there as a reference document at this point. If you do have additional feedback, as I said, it's a live document. We're just going to keep updating it each year at this point, rather than locking it in as a 5-year. To keep it fun. Any particular questions or comments or thoughts? You're going to hear a lot of the streams over the next day or 2.5 days.
I've just been trying to understand the lay of the land so far, and so looking at everything useful, figured out understand a lot better after this.
One of the things we've talked a lot about over the last 5 years that I've been on the SRB is the links between the different programs and how addresses critical uncertainties that they see stock assessment and this as well. And just making sure that those links are clear and that position happens across the research areas, not just. I think that's clear in the priority lists.
Any questions online or comments from Anne Marie or Pete? Seeing any? Okay, now we can get into the real material of the meeting. So if we could have. Let's bring up the agenda again, please.
Dave, so sorry to interrupt. This is Courtney online.
Courtney.
Just want to note that last speaker there, it was very hard to hear. I'm not sure if folks were a little bit far from the microphone, but just flagging some audio trouble. And sorry, I was also having some mic trouble there earlier to introduce myself. Thanks for taking a moment. My name is Courtney. I'm just joining on behalf of DFO's Fisheries Resource Management and Negotiations team. Thanks.
Thanks, Courtney. Can you hear me now?
That was a bit better, yes.
Yeah.
Oh, it's not the owl.
No, yeah, the owl's just the video.
Okay.
So project yourself up. Okay. Moving on, I'm actually going to go down to the schedule. It's going to be a little bit easier to follow. So the first is within the research stream 4.1, the biology and intelligence. So the presentation for paper 06. You're going to present direct from your computer, just
Yeah, I'm going to try.
Yeah.
Perfect, thank you. Well, good morning. I'm happy to present our updates on the biological and system science research activities. For Mike's benefit, we have contemplated within our program of research and integrated monitoring, we have 5 main research areas that we're working on. One is migration and population dynamics, and I'll speak to those in sequence. The second one is reproduction. The third one is growth. The fourth one is mortality and survival assessment. And the fifth one and last one is issue technology. I usually start each update on the respective research activities with a summary slide of previous work we contemplated within previous research plans, and then lastly highlighting in this column here. Actually, I can't control. Just pointer. So this is the current program and research and integrated monitoring, and highlighted in red are the activities that we're conducting right now and the ones that we're going to be updating you on. As Olaf will recall, we've been working over the last couple of years and presenting results on establishing a baseline of genetic diversity with the purpose of delineating stock structure at a fine scale. So obviously the implications for management is to determine whether there's a need for separate assessments for certain components of the stock, and whether there's a need to potentially revise structure of future stock assessments, knowing that currently the stock assessment manages the same stock. Andy will provide some updates on the results of this project that is almost completed. And what I will do here is just highlight the things that we're going to be commenting on. First is an update on the fact that we're actually writing 2 manuscripts at the present time on this work. One is in fact describing the genetic population structure that Andy will speak to in just a minute. This paper is in preparation. And then we have a second paper that this is new for the SRB, and this is regarding the development of a new bioinformatic method for analyzing low coverage whole genome sequencing data. So this paper is actually a standalone paper on the development of a method that was implemented in our analysis of population structure. So this paper is actually now in its very final drafts. We're about to submit it to a journal, and it will speak to that, and he will go into a fair amount of detail into the work that he's put into the development of this method and its validation. Okay. Well, I'm also going to provide later on an update on the development of an epigenetic clock for Pacific. Okay. So with that being said, Andy, you want to go ahead?
Yep. All right. Thanks, Giuseppe. So yeah, so like Giuseppe mentioned, today I'm going to be focusing on describing the development and validation of a novel method for estimating levels of genetic differentiation from genotype likelihoods. But before we get into a lot of detail there, I thought it'd be nice to to start with a little bit of background on kind of the larger context of the project that this method fits in. So this method was used as part of our analysis of population structure among spawning groups, utilizing low coverage whole genome resequence data. And there are some caveats that kind of go along with low coverage whole genome resequencing data that we'll get into later. But the samples that we're utilizing for this project are shown in the map here. They were all collected from spawning groups. So that means they were collected on or near known spawning areas during the spawning season, which is the wintertime for Pacific halibut. These samples were collected between the years of 1999 and 2020 from 5 different areas shown in the map here. That's British Columbia, the Central Gulf of Alaska, Bering Sea, and the Central and Western Aleutians. And we submitted a total of 739 samples for sequencing, targeting approximately 30 male and 30 females from each sample collection. So that's each combination of year and area. And so the figure you see on this slide are the first 2 principal components of a principal components analysis we conducted using this data set. So Each point represents an individual fish, and each fish is colored by the area in which they're sampled in. And you can clearly see there's a lack of structure observed here. So the results of this analysis essentially provide evidence to suggest that there's a lack of genetic structure among spawning groups of Pacific halibut in IPHC convention waters. So as far as the kind of motivation for developing this method is concerned, from the data analysis perspective, is that low coverage whole genome resequencing essentially creates a sampling problem. So as you generate genetic data, the goal is to essentially sample both alleles, the allele that's inherited paternally and maternally. And so when we talk about sequencing depths, essentially think of the number of times you sample those alleles with your sequencing technology. And so at lower depths, this creates an element of uncertainty about an individual's genotype, because you may not have actually sampled both of those alleles. And so there are kind of frameworks to work in this, specifically utilizing genotype likelihoods. And so essentially what a genotype likelihood is, it's the probability of observing a specific set of sequence data given the specific genotype, or given a specific genotype. And while low coverage whole genome resequencing is a newer high resolution method, there are a limited number of tools that are actually designed with genotype likelihoods and work with this uncertainty. And so I put together kind of a quick animation to sort of provide a visual to the data generation process and how this plays out in terms of genotype likelihoods and how you might actually make a determination on those individual genotypes. So here we have, so in this scenario, we're trying to sample a heterozygous individual, so an A allele and a C allele. And we have this reference sequence here. It's just a bunch of A's, and the SNP that we're trying to sample is bolded and underlined. And you can see at this point, since we don't have any sequencing data, this individual has an equal probability of carrying any one of those three genotypes. And at this point, we can't make a call on the individual genotype. So let's say we go ahead and generate 2 sequence reads, and just by chance, we happen to sample 2 C alleles here. So the genotype likelihoods are updated to reflect that. And if we were to call this individual genotype, this data alone, we would call that individual as being heterozygous for the alternate allele. So now we start to generate a little bit more sequence data. And you can see we have sampled that A allele. And again, those genotype likelihoods update to reflect that. But in this case, it's very hard to distinguish that A allele is maybe the result of a sequencing error, or if it's actually the true allele. Now, if we go ahead and generate more data, here we have an equal number of A's and C's that were sampled. And so we can actually go ahead and call that individual as a heterozygous individual. But if you look at the genotype likelihoods, there's still a small bit of uncertainty that's associated with those individual genotypes. And I guess, yeah, I should also just preface that when we say low coverage whole genome resequencing, we're essentially talking about, say, like 3 to 5x, so somewhere in this range. So a large amount of uncertainty associated with these individual genotypes. So on this slide, I guess I want to discuss the motivation for the development of this method in terms of its application within our analysis of population structure. And so one of the main questions we had in our analysis of population structure was, can we actually assign individuals back to the location that they were sampled from using a subset of potentially infected SNPs? And so the short answer to that is no, we cannot. If you look at these box plots here, these summarize the overall levels of assignment accuracy we used when we conducted these tests for the different test training split combinations that we tested out and the various SNP panel sizes that we evaluated as well. And so in all cases, overall assignment accuracy was low. It was less than 40%, even when you use a very large SNP panel that consists of 25,000 SNPs. I mean, that's a very large SNP panel that is probably kind of on the limit of what would be, what you would even consider doing if you were going to construct a panel to do this in like a production setting. So within this, this method is specifically focused around selecting the SNPs used for this panel. So our method estimates FST, which is a measure of genetic differentiation, that we then use to essentially rank those SNPs and make our selections based on that. And so what we needed was a way to repeatedly select SNPs for use in these assignment testings, because we wanted to test out a number of different SNP panel sizes and training sets designs to see if we could, optimize those levels of assignment accuracy to some degree. So as far as the method itself, we're calling this FSTGL. It's written in the NIM programming language. And so if you're not familiar, NIM essentially is translated to C code before it gets compiled to machine code. So it's very fast out of the box, so to speak. And it leverages the HTS NIM library for parsing standardized file types associated with high throughput sequencing data. So it fits nicely with existing tools that are designed to, query and subset these various files associated with genomic data. It uses OpenMP to implement multi-core processing for some sections of the code to further improve performance. And at the core of this, it uses an expectation maximization algorithm for estimating allele and genotype frequencies directly from genotype likelihoods. And then these are actually used to obtain those estimates of FST. So we also did substantial validation for this method using both simulated and empirical data. And so as far as the simulations are concerned, we simulated we conducted individual-based genetic simulations based on a Wright-Fisher model using the software SLIM. And we simulated 2, 3, and 5 population scenarios under 4 different migration rates, ranging from no migration up to 10% movement each generation. And this was simply done to just generate FST ranges from 0 to 1. So we wanted to generate the entire range of possible values of FST here. Then using those genotypes that were sampled directly from the simulation, we then went ahead and simulated sequence data for each of those individuals. We used a different simulator designed to simulate sequence data using an Illumina NovaSeq error profile, which is the same sequencing platform we used to generate the raw data for our population genetics project. And we simulated the sequence data up to a depth of 20x, so 20 sequence reads on average throughout the genome. And then we downsampled that data to various depths of sort of different categories. So the first two, 0.1 and 0.5x, this would be considered like very low or ultra low coverage. So a lot of uncertainty there. 2.5x and 5x, that's kind of in the low to medium range, which is what our data set, I would consider our data set to be in. And then 10x and 15x, which are higher levels of coverage, which you might expect that uncertainty to become negligible at that point. And then finally, we compare the estimates of FST that we obtained using the genotypes that were sampled directly from the simulation in our new method. What would happen if you just used hard-called genotypes that were obtained from the low-coverage sequence data? And then estimates obtained between ANGS, which is an existing, fairly well-used likelihood-based method. And this tool can only produce pairwise comparisons between 2 populations, so we could only make the comparisons in those cases.
Sorry, Andy, could you explain what hard-called genotypes are? Is that where you're just picking?
Yeah, you're just exactly.
So you're not using
Yeah, no probabilities. It's just, you assume that there's no uncertainty associated with like the sequencing process, right? Or I guess the sampling of those 2 alleles, right? So then we also validated this method using some empirical data that we've generated as part of our population genetics study. And we chose the example of, and we did this to provide more of like a real world use case type of scenario. And so the example we selected was the identification of a sex-associated region on chromosome 9 in Pacific halibut. We chose this because this analysis was completed and published as part of the genome sequencing effort using different techniques, pooled sequencing. But nevertheless, those results are published. And the samples that we were able to essentially recreate the same sampling design using a subset of samples from our population structure study that was done in this paper. So that's we used 30 males and 30 females that were collected on Portlock Bank in 2018. And as far as the sequencing depth of this data set that we're using here, that's about 2.9x on average. And this data set consists of about 240,000 SNPs. And in this case, we're only comparing estimates of FST obtained with our method to that of angst, which is the alternate, existing likelihood-based method. So as far as the performance of our method is concerned in comparison to other methods, we compared that using RMSE and median bias along with the 2.5 and 97.5 percentiles. And so these figures are laid out going from left to right, so that the columns are a 2 population scenario, a 3 population scenario, and a 5 population scenario. And then going from top to bottom, that's the sequencing depth. So the top 2 rows are the kind of ultra low coverage scenario. The middle 2 rows are kind of the low to medium coverage scenario. And the bottom rows are where you might expect high coverage scenarios, where you might expect that uncertainty to drop substantially by when you're using hard called genotypes. And essentially what you can see here is that, sorry. So the different lines, so our method is represented by the blue line, and the orange line represents what would happen if you used angst, which is again another likelihood-based method. And the gray line is what would happen if you just went ahead and called genotypes directly from the low coverage data. So you can see that the likelihood-based methods start to perform pretty well, even at very low sequencing depths. And our method is, in the case of pairwise comparisons, is comparable to other existing likelihood-based methods. Kind of the main benefit to the method that we've developed here is that it's a lot faster. So we observed anywhere between about 69% and 94% reduction in wall time. So that's, the real time that the computer is churning to produce these estimates compared to angst, which again is the kind of really the only other likelihood-based method for producing these estimates currently that I'm aware of.
Is the RMSE on those the RMSE for estimating FST or something else?
Yeah, exactly. So we just take the right. So we take the value of FST that's obtained using the simulated genotypes, right? So observe without any error and subtract that from the estimates. And that's calculated within bins. We bin the data to be able to do that. So these are, I think, binned by 0.5. But yeah, essentially it's the kind of the error around what the true value should be. And the y-axis is different for all of these because we wanted to, I guess, get a handle of like subtle differences between the methods. So I guess, yeah, moving on to the empirical example, the analysis of the empirical data. So the figure at the top here, this is called a Manhattan plot. And so this shows sliding window estimates of FST along chromosome 9, which is the chromosome that we identified to be sex associated for the species in that paper that I mentioned. And so in this figure, estimates obtained with our method are actually in yellow and angst is in blue. And you'll see we identify a nearly identical signal between both methods. Those dashed lines you see represent the sex-associated region that was identified in previous analysis. And we're essentially recovering the same region with these analyses as well. But we also were able to kind of generate a new piece of information here. So this method also produces estimates of heterozygosity. And so the figure down below is heterozygosity levels for individual SNPs across chromosome 9 for females on the top and males on the bottom. And you can see that there's a large number of fully heterozygous SNPs detected in females within this region of the genome. Keeping in mind that Pacific halibut utilizes ZW sex determination system. So females are actually the heterogametic sex in this species. And so, yeah, just interesting new piece of information that, supports kind of the results of the previous analyses that we published on. And finally, in terms of like real world performance, so this bar chart shows the total wall time of the various steps required for each of these workflows. So on the left is the difference. So this describes the ANGS workflow. So there are 5 different steps here. And to produce this analysis, that took about 32 minutes. And on the right is the workflow associated with our method, which took a total of 20 minutes. But that yellow bar you see there is the genotype likelihood estimation step, which is done prior to the execution of our method here. And that purple bar represents the runtime of our method, which I think it took only about 11 seconds to analyze this data set. So, and that's really what enabled us to do those resamplings around the assignment testing, is because we only had to do that genotype likelihood estimation once. So it kind of opened up the door to conduct that analysis in our analysis of population structure. And so, yeah, like Josette mentioned, we're kind of on the final round of polishing a manuscript for this. We hope to, or the plan is to submit it to the Journal of Bioinformatics as an application note, so that, the method and all the validation is published separately from the analysis of population structure, to keep things clean. So, yeah, that's all I have. If there's any questions or move on.
Andy, could you give us a sense for how constraining the runtimes are? It seems like a big benefit here.
Oh, yeah.
Speed. Is that, are you talking about, runs that take a day on the computer?
Yeah, definitely. I would say yeah, so in this example, right, you're talking about 30 males and 30 females, which is a fairly small data set. Compared to we had 700 individuals in our entire data set. Yeah, I mean, it probably would take, I'm trying to think, because it was, I kind of left behind the ANGS stuff along, after we got this method going, but it was probably at least a week, because you're doing all these pairwise comparisons, right, among, with larger numbers of samples. And this is only a single chromosome, there are 24 chromosomes in the genome. So yeah, it's quite constraining.
So for that example, it went from a week down to
basically, yeah, probably like a day. I mean, so yeah, 11 seconds. And, you only have to do that genotype likelihood estimation step once. And yeah, so it opens the door to do, resampling type of things with this type of data.
Okay, thanks, Andy.
Yep.
Yeah, so moving on, what I'd like to do is just to provide you with an update on this new project that we started. In fact, last year it was reported and introduced for the first time in the SRB 27. That was last September. We do have a few updates to report, but before we do that, as a summary and a reminder of what this entails.
Sorry, one question back, Brandy, before we move on. With the shallow reads, can you also get the sort of information that you might use for relatedness estimates if you were going to explore something like close kin marker capture or something?
I mean, that's a good question. So there are programs out there designed, to produce related estimates between individuals using genotype likelihoods. So yeah, there are methods out there that exist. Well, I mean, whether or not those are suitable for close kin kind of things, I guess, is another question. But yeah, there are tools out there to do that. So
Yeah, so I'm going to just provide a brief update on the development of an epigenetic clock for Pacific halibut. This is an alternative method for aging. One of the advantages that is a non-lethal method that can use genetic samples. The genetic samples that we're using for this project are fin clips that we routinely collect and both from our fisheries independent satellite survey and from our commercial samples. As a reminder, epigenetic clocks are molecular aging clocks. So those are machine learning algorithms that are trained on epigenomic data from samples that reflect a wide range of ages. So epigenetic clocks use as input genome-wide methylation features at cytosine, guanine, dinucleotide pairs in regions of the genome that are GC rich. So what it's providing is a proportion of DNA molecules that are methylated at each CPG site for each individual. This is used as input. So the tables are generated, and this is a table drawn from a review of the development of the human epigenetic. Shows different individuals, in this case, 10,000 individuals with known chronological ages, shaded them, and the percent methylation status of each CPG site that is identified in the genome. In this case, up to 850,000. So these tables are actually used in age predicting models, and probably one of the most commonly used for the development of this kind of clocks are elastic net penalized regression models that rely on the weighted average of methylation across a subset of CPG sites. So here you actually select those CPG sites that have the highest association with age. So in order to do this, there's a test data set that is used to validate and test the performance of the epigenetic clock. You derive Pearson correlation or median absolute errors. So in our case, we set out to develop this epigenetic clock using the following sequencing technology, and that is the reduced representation bisulfite sequencing. With bisulfite sequencing, you actually are able to convert unmethylated cytosines to uracils, whereas methylated cytosines are not altered. The sequencing process generates basically what we call methylation calls. So the percent methylation status of each CPG site, and that information, those data are fed into the age predicting model that eventually will generate an epigenetic clock that will be a relationship between the predicted epigenetic age and the chronological. So this project, as I said, is relatively recent. We started last year and received funding from Alaska Sea Grant, and it's done in collaboration with Alaska Pacific University. And there's a master's student whose project is the development of this clock, who is in her second year now. And she's getting her master's degree from Alaska Pacific University, our collaborator. So these are the samples that we drew from to conduct this study. So we selected a wide range of age samples. So those are samples, fin clips from fish that were aged twice by 2 independent readers, so that the ages were as correct as we could get them. And from that sample collection, which spanned the years from 6 years of age until 30, we selected 10 individuals per age, 5 males and 5 females. So a total of 249 samples. These are the in this map you can actually see the where the samples came from. They actually span the entire convention area. We have samples that were collected from our fisheries independent satellite survey between the years of 2001 and 2021 and 2024, and obviously they're mostly concentrated in the areas where the survey has been focusing on all those 4 years. And that's, as you can see, is in regulatory areas 2B, 2C, and then 3A and 3B. So basically covering most of the convention area, but focus on those areas. So, but we also call biological regions 2 and biological regions 3. So this is a representation of the different samples by age that we have, and we actually succeeded to get 5 individuals, 5 males and 5 females from each ages, from ages 6 to basically 28, 29. We're slightly under our target for age 30, which is hard to get individuals aged that were so old. But I think we had a fairly comprehensive data set. So these are the results from the sequencing that we've done. So far we've sequenced all 249 samples. Those samples were sequenced in 6 different pools. About 41 to 42 samples were pooled and then sequenced separately. So we have the results from those 6 sequence pools. The average PCR duplication rates are acceptable. We have an average of 43%. What's important is the 2 controls, so that the average of the methylated controls is under 2%. We actually get a 1.58%, and then the average of unmethylated controls, which you want to get as close as 99 or 100%, is above 99%. So those controls, positive and negative, are actually really good. And the total amount of data in terms of the number of deduplicated reads is 1.6 billion reads that we have to process now. So we have an in-house bioinformatic platform that Andy developed that we're currently processing those samples. So they're basically now in the QC and read adaptive trimming and genome alignment step. So that's as far as we have gone so far. So sequencing has completed and currently processing this data through our bioinformatic pipeline. And I hope to be able to present some results in the September meeting. Are there any questions regarding this part of the work?
Yeah, Joseph, can you remind us how specific those regression models are? You developing separate regression models for 2 sexes or for different years or different regions? Or is it a single regression model?
It's just initially we're going to try to do a single regression model, including both males and females. Yeah. So the 10 individuals per age.
And we had talked at the last meeting about looking at out of sample cross validation. So
That's what's coming up next. Yeah, yeah. Because this was the request from the SRB, right? So that request, there was a recommendation. The recommendation said that evaluation of epigenetic aging be expanded from random selection of cross-validation samples to include testing out of sample interannual predictive performance. That is, how well can an epigenetic aging method train on data from one set of years predict age of individual sample and other? So as a reminder, the data set that we have for the training and test model is the 249 samples collected from the Fisher's Independent Senate Survey from the years 2021 to 2020. So for this, what we've done is actually we've selected additional samples from other years, basically from 2017 to 2020 from our commercial sample. So again, these were samples that were double aged. So the ages, the chronological ages is fairly accurate. And these are the distribution of ages on the right-hand side for females on the top and males on the bottom. So as you can see, most of our samples actually comprise the ages between 6 and 20, which is really what you encounter in the tissue. So if this was to be applied to age fish coming from commercial samples, then we have a good range. So those 46 samples were processed. Genomic DNA was isolated. Libraries were constructed from all those individual samples. They were pooled into one single pool, and that library pool has been sequenced. We have already the results. That's very recent, and we're currently processing that data. In the table on the bottom, you can actually see the distribution by regulatory area. So 46 samples. Except for area 4C, it's fairly evenly distributed among different regulatory. So that pretty much completes the update on the development of the epigenetic clock. If there are additional questions?
Yeah, just one additional potential benefit of having this would be for difficult to age individuals, and difficult to age from otoliths. So at what age do they become difficult to age from the otolith? It's above, say, 20 years old, hard to distinguish, or at the annualized still pretty clear.
Maybe Ian knows that question. I'm not sure. I know that older ages are particularly difficult to age, and I think it's above 30 because all the rings are kind of
Above 30, so not really relevant.
No, I think up until 30. I don't know. You guys correct me if I'm wrong.
So we have a plot that shows that, the distribution of kind of
Yeah.
error, so it's just increasing as the age goes older. So it's a kind of
Pretty loose.
stepwise increase, but
Not a big real
there's no
threshold
like
there.
a specific age where the settling the age becomes difficult. So we did do that experiment where we had multiple ages. We get the confidence interval with the aging, and that confidence interval is increasing as the patients age up older. So
Sure. The other potential benefit is cost. So I can't remember, I think we talked about this last time, but did you look into trying to figure out what the costs are in terms of staff time and materials for the otolith-based aging versus?
Yeah, we did talk a little bit about it, and we thought a little bit, but one of the outcomes of this clock development is the identification of a subset of CPG sites that are so. So it's impossible right now to say how many CPG sites we're going to be able to use for aging. From the literature, it could go from 18 to a couple hundred, depending on the species. So that will mean that the panel, and that's going to be probably most likely a PCR-based panel, that will be used. So depending on the size of the panel. So it's very hard to say exactly what's going to be the cost benefit, because we can't apply costs that are incurred in development of the clock to the costs that are going to be incurred by the actual panel that's going to be used for aging. So that's a discussion that we're going to have. Sequencing technologies are getting cheaper and cheaper. So that's another consideration. But and the method for developing that panel, which is most likely going to be a GTC panel. That could change dramatically. So hopefully, as we get along and we get better information on which are the number of highly age-associated methylated sites, that you'll have a better idea on how much that. What are the costs, compared to the AI-based method or traditional aging.
So at this point, at least, it seems like the major benefit might be being able to age samples that you can't cut the heads open on. Whether that's like research samples where you're releasing them live, tags on them, or commercial samples where you can't cut them.
And even developing down the line a combined method for sexing and aging.
It's the one fin clue.
Aging, we're sexing more than 10,000 individuals every year to landing already in a routine manner.
Yeah, this is super cool. And one of the things that the student might consider doing is, given the whatever different years they have, they can slice and dice the data differently to have their training data set and then validation data set and look at it as well with this other out of sample data set to try and get some idea of whether this is varying spatially or temporally. And that'd be, once you have all the data, those will be really straightforward analyses to try and do to get a handle on that sort of classification accuracy.
Yeah, I think I agree. I think the validation step is going to be really interesting.
Because I was talking with a colleague the other day about the CPG sites that you throw out in the analysis. And he has been working on seeing whether those are stress markers, because they don't build up consistently through time. And I guess there's some indication from zebrafish that they might be markers of like cumulative lifetime stress.
Right.
Is that something that you guys have talked about doing with this? I mean, it seems like you'll have the data.
Yeah. Yeah, I mean, that's one of the advantages, exactly. One of the advantages of this method is that you're actually looking at biological processes. That you can infer some information regarding longevity and stress, and I'm aware of that literature. So that's something that would be potentially interesting.
Yeah, that could pan out in really interesting ways. If you could develop a lifetime stress marker and ask, for example, whether individuals that have experienced certain oceanographic events, pre-heat waves or something like that, are showing their incidence of stress markers.
Yeah, we have a tremendous amount of data of fish that have been recaptured. We record prior hook injuries, and so that's another stress in the fish. Yeah, definitely. There's a huge amount of data that can be mined in addition to actual age.
Yeah. You have tissue samples from all of the tagged fish, right?
Recent years, yeah.
Yeah, I think the big question is what sort of stress this is. Is this a marker of thermal stress, nutritional stress, catch release stress?
Even if you're just seeing changes in stress over time, though, that would be cool, even if you don't know exactly what it's related to.
Spatial. So yeah, we'll figure that out.
We got size at age and also weight at length for all these fish as well in recent years. We could look at condition as well as growth expression.
Yeah,
lots of cool stuff there. The big gap in our historical tagging is that we have length at tag release and often length at recovery and not age. And because of the spread of size at age for halibut, this is basically nothing for age. So all of the aspects of movement and that are related to age but size are completely confounded in real data. So this tool unlocks that in the future. We now have a ability to at least to refine down to a really reasonably precise age range. That's huge for future tagging studies. We have 500,000 tags we've released and 50,000 recovered, and we know very little about the sex and age dynamics of those tagged fish because we can't recover it from the size data. Big challenge.
Well, not just future tagging studies, but reinterpreting past tagging.
In recent years, yeah. We didn't start collecting fin clips until 2017.
17 or 18?
Yeah.
17. Definitely.
So, almost 10 years now. We have 100 years of data.
Both in our fish and from commercial samples.
Cool. So exciting.
Okay, so if I may, I'll move on to the next area, that is reproduction. And just to give you a little bit of background, where we're coming from. In 2017, we started investigating the reproductive biology of city halibut, focusing mostly on females, and we conducted quite a bit of work characterizing the reproductive development. A few important outcomes came out of those studies that were published in these papers that you see listed here. One was the type of fecundity the Pacific halibut has. This is crucial for what Colin is going to be explaining in a minute. Pacific halibut has determinate fecundity. So it's a type of fecundity that is suitable for the methods that we're going to be trying to implement to estimate fecundity in the species. But most importantly, for what Colin has been working over the last few years is the revision of maturity schedules based on histological data. Up until last year, all the maturity schedules that were incorporated into stock assessment were derived from visual macroscopic staging of ovaries, and we set up a detailed method to classify the developmental stage of particular females based on histological data. So we were completely revised our maturity schedules based on the histological data. That's what Colin's going to be describing, and we've also started to set our gears to begin fecundity assessments. Over the last few years, we've been collecting a number of samples for histology that require the collection not only of the physical ovarian sample, but the weight of the ovary for the method that we're going to be using. And this is, again, something that Colin will explain. The histology-based maturity schedule that is completed was presented to the SRB in its completed form, I think, in the June 2025 meeting. So that was that must have been SRB028. That was the detailed description of this study that is now basically being written in a manuscript form for publication shortly. So those are the 3 years of spatial coverage of maturity data that we generated between 2022 and 2024. So Colin, you want to go ahead?
Yeah.
Can you?
Yeah, they just made me presenter. Thank you. Yeah, so I'll just, the first couple of slides that I have kind of provide a little background of what was presented last year, so you can get an idea of the work that has been done to update our maturity ogive in our current stock assessment. So as Joseph alluded to, from 2022, since 2022, we've been collecting histological samples on our fish independent satellite survey every year. And so our initial data set was a 3-year time frame from 2022 to 2024. And what we did was we created a calibration between our visual estimates and our histological estimates to update our visual maturity timeline curve through time. And so we were able to make a longer historical curve dating back to 2002. And 2002 was chosen because that's when the IPHC changed their primary aging method from mostly surface age reading to break and bake. So we were able to create a timeline of visual maturity data from 2002 to 2024. And so using that calibration between the histology and the visual, because every fish that has a histology sample was also a visually maturity estimated in the field on the survey. And so what you see here on the figure on the right is that red line was our previous maturity estimates for Pacific halibut. And that data was reflected in just 2002 and 2003, taken from regulatory areas 2B and 3A. And that has been implemented. That curve was initially implemented in the assessment, I think in 2006. I think that's right. And so we have been using that historical curve from 2006 until 2024 assessment. So it hadn't been updated in a really long time. And so what you see here on the figure is that green line is now our calibrated visual maturity estimated for a coastwide curve. So basically what we did was we estimated biological regional estimates and created coastwide curves by year dating back to 2002, and then averaged over time to create this green curve. And then what you see is that the current estimate is about 0.6 years for an A50 or age at 50% maturity value. It ended up being about 0.6 years younger compared to the historical curve that was implemented in 2006. And then also another thing that we did was our previous estimates using just visual data estimated that no female younger than age 8 was mature. And we found that using histology, that actually ended up being age 7. So the old curve was truncated at age 8. We've now truncated the new curve at age 7, assuming that there's no females younger than age 7 as mature in the population. And then, so this new coastwide maturity ogive was implemented in the 2025 stock assessment. And if you're interested in Ian's sensitivity analysis for that, go back to his document from SRB028, and he provides all the documentation on the impacts that this curve had on the new spawning stock biomass estimates for Pacific halibut.
It's surprisingly important, given how small those differences are there, but that's where so much of the biomass is.
Yeah, because right now, currently, our population has a very young population, and so younger ages, it's actually making a pretty good impact on years where the population is showing young fish.
Of course, it occurs on both sides of the numerator and the denominator in our reference points. So small changes add up. We also did a sensitivity to the truncation because the model was predicting there would be some mature fish at ages 5 and 6, and there's a lot of fish at those ages, but we were a little suspicious that they were actually mature because we're just fitting a parametric shape. And so we did a sensitivity to that, and that curiously didn't have a huge effect. Those fish are small enough, and I guess the cumulative reproductive output of a small fraction on small fish, even though there were a lot of them, didn't have a huge effect on the assessment. So that one I was a little worried about early on because we didn't really have a clean way to get at that without having a lot of samples of those young fish, and it turned out that was not good. But again, we did a sensitivity in the assessment to see, and we ended up using the truncated version following the same logic as before, that just because the parametric model was suggesting maturity down there, we'd want to actually see at least a single actual fish mature at those ages before we included them in spawning biomass.
Yeah, one thing that I didn't note was that, so previous maturity estimates were just using GLIMs. We compared GAMs and GLIMs, and the GAM actually is a better fitting model for Pacific halibut, provides a little bit more flexibility in the model. So that's what we're using, our GAMs. And also it probably didn't make much of an effort, but you do much of an impact on the assessment, but you do see that there's a little bit of drop in the new curve at slightly older ages. And that's not surprising because histological methods allow us to determine immature fish at older ages better than field classifications can. And that's something that's pretty common among a lot of fish species.
I'm trying to remember where I had this discussion now and even what species it was about, but it might have been with blue crabs actually, but there was a question about whether some fish that are histologically identified as being mature are actually going to spawn.
Yeah.
And so I'm curious if you.
Yeah, so that is a great question, and that's skip spawning is still an avenue of research that we're doing here. For our maturity estimates in the assessment, we have decided to just go with mature classifications and using that in the assessment. We are leaning more towards using potential skip spawning rates or reduction in potential egg output more towards our fecundity estimations. But that is something that we're definitely still looking at. All of our histological samples, we are classifying rates of atresia reabsorption of egg cells. And so we have that data moving forward, and it's just a matter of where we want to ultimately implement that. One of the things though that we have seen histologically is that Pacific halibut tend to be a very robust species, and we don't see very high levels of atresia in Pacific halibut in particular. Now, obviously, that could change if you have specific environmental impacts, like if we have another warm blob in the Gulf of Alaska, that could potentially change things. But that's something that we're continuing to keep monitoring over the long term.
Yeah, I was thinking more so though for the younger fish, rather than necessarily skip spawning later on, but if those younger fish are getting eggs that look mature, but they still might not actually spawn.
Yeah, so we do, that's another great question. So that would be more abortive maturation, what they classify as abortive maturation. So a fish that's immature, that's trying to mature for the first time, but ends up aborting. I would say that when you get to vitellogenesis of Pacific halibut, so the initial development of eggs for that year, a lot of times there's a distinct breaking point with Pacific halibut where the fish is only in what we would consider to be late primary growth or cortical alveolar stage, where they haven't start hitting vitellogenesis yet. And then you'll start seeing immediate atretic cells. And so you can see when you have, like, let's say 7-year-old or an 8-year-old, you can a lot of times specifically identify that. But a lot of times what we've seen with Pacific halibut is once they reach vitellogenesis, they're progressing along. They're not stopping. Yeah.
Cool. Thank
And so to give a little bit more information on temporally and how maturity rates have changed over time for Pacific halibut. What you see on the figure on the right here is our coastwide. And so when I say coastwide, I mean across all IPHC convention waters, basically, because there's just one maturity ogive in the stock assessment currently. We've actually broken this down by year. So from 2002 to 2024, it's a little bit hard to see kind of the differences, but you see that starting the black, kind of more darker lines are kind of in the middle of the curve there. And you can see that there was a distinct shift to the right, or older maturing females. But then in more recent years, there's actually been a shift to the left to a lot more higher proportion of younger maturing females. And to look at that a little bit more closely, we didn't present this figure last year, but I thought it'd be nice to kind of see. What you see here are the two different lines. So that dotted line is the raw visual maturity estimates from the field. And then that black line is the calibrated visual estimates using the histology data. And what you can see here is there's kind of been two distinct temporal shifts with females. From 2002 to 2016, there was a slow rise in the age at 50% maturity. And then after 2016, there was a distinct drop off to about 2022, where there was a large decrease from about almost an A50 of 12 all the way down to almost 9.5 years old. And so this is definitely something that we're looking to hopefully do a little bit more analysis on to see maybe drivers of this and how this potentially is directly impacting the overall population. One thing that we could definitely look at is, are shifts like this in maturity impacting potential size at age in females in the population, and also looking at environmental drivers, obviously looking at, because, you know, in particular, like what was it, 2013, 2014 was that big warm blob in the Gulf of Alaska, and how potential impacts that might have had on the overall maturity rates of Pacific halibut. So this is something that we're definitely going to be looking to see whether or not the leveling off here stays consistent or starts going up or down in subsequent years.
This is super cool.
Yeah, it's
excited to see this.
Yeah, it's really neat to have such a long time series like this and be able to look back. Because we have, obviously the aging methods has changed, but we have visual maturity data back to even like back to the 60s and 50s. So it's really neat to be able to see historically how these shifts change and their potential overall impacts on the population.
One of the tricky questions that will come up in criminal variables is just figuring out the timescales that are relevant. So
Yeah.
probably not just the year.
Yeah,
maybe what
yeah,
the-
yeah, yeah, exactly.
Something about what that cohort experienced.
Yeah.
Up to that.
Okay, so I can, that's all the kind of updates that I have on the maturity estimates. We're still going through the 2025 histological data. So I'm hoping to potentially, hoping to be able to present that at the September meeting to give a little update on whether or not we're seeing current trends in our maturity estimates. So now I just wanted to give a little background on where we are and where we're heading with fecundity estimation. So our current assumption in the stock assessment is that there's an isometric relationship with weight, body mass, and egg output for female Pacific halibut. And the ultimate question is whether or not there's potential hyperallometry and those types of effects in females. That's a big question that a lot of stakeholders are asking. And so we're really interested in really diving into that question. And so like Josette mentioned, we have determined that Pacific halibut have determinate fecundity. And so because of that, we're able to specifically use what's called the autodiametric method. So previously, a lot of times when people were doing fecundity estimations, it was you take a subsample of the overall gonad and then you physically count eggs. The autodiametric method, what it does is it allows you to take a fresh ovary weight in the field. So we do that on our set line survey. Then you take a subsample and you preserve that subsample in formalin, and that comes back here to the office in the lab. And then what you can do is you can create what's called the autodiametric curve, which is what you see on the figure on the right here. And what that is on the x-axis, you have mean oocyte diameter, and on the y-axis is oocyte density, or basically eggs per gram of tissue. And you can use, basically you take a weighted subsample, you get a mean oocyte diameter, you'll be able to create that curve, and then you're able to extrapolate that out using the total gonad weight to get a potential annual fecundity for that individual female. And the benefits of the autodiametric method is that once you build this autodiametric curve, you're basically able to skip the weighted subsample portion and able to just take a small subsample of about 200 to 300 oocytes, get a mean oocyte diameter, and then be able to use your curve to get a oocyte per number of oocytes per gram of tissue, and then extrapolate that out using the gonad weight. So once the curve is built, you're able to process samples a lot faster. So that's kind of the reason why we're using this type of method. And so starting in 2023 is when we started using our set line survey to actually go out and collect fecundity samples. So in 2023, we specifically just collected samples in biological region 3, which is the Gulf of Alaska, which is what you see with the red dots. And then 2024, we were able to collect samples in the northern part of region 2, as well as the Bering Sea. And then in 2025, we were actually able to collect samples across all 4 biological regions, which was great. And then also in 2024 and 2025, we started a special collection, which was trying to collect samples for later developing females to help us build our autodiametric curve, because we felt that within our time frame of the set line survey, which is usually about Memorial Day to Labor Day, we wanted to try to get some females that were a little bit later developed. And so in 2024 in biological region 2, more specifically the Charlotte Charter region, which was kind of the northern part of British Columbia. We collected, we set out and collected some fecundity samples for females that are greater than 90 centimeters in the months of October and November. And then we did it again in 2025, but a little bit earlier in the months, I think it was like late July through the month of August. And I'll show you a little figure, I think, on the next slide. So those are kind of the samples that we've collected. We have hundreds of fecundity samples now across all 4 regions that we're gearing up this summer to be able to start processing. So in more particular for that special collection in region 2, what you have here is the bar chart on the right. So in 2024, we were collected our samples in October and November. And in 2025, we did July and August. And then all those samples have now been histologically staged. So we can see the actual developmental stage of those females. And in 2024, this is actually the latest we've ever sampled females in biological region 2. We've never seen development of females this late for region 2, which was great. And what we see is that most of the females are in very late vitellogenesis or early oocyte maturation, which is the germinal vesicle migration stage. And then also in 2025, we see a lot of mid to late vitellogenesis females. So our sample collection was really good timing because now we're able to use all of these females to help build that audiometric curve. And also, since you brought up that question earlier regarding potential skip spawning or even potential late developed females that might not progress to us, you can actually see that on the bottom there in October and November, there is a very small proportion of our samples that were only in VTG1 and have not progressed past 10. So those females there could be a potential indicator of potential skips. And the level that you see, so it's you're probably talking rates that are less than 5% so that just kind of gives you an indication of the number of females that you might see that maybe have hit the very early vitellogenesis and then decided I'm not going to do it. Like I'm gonna abort that year. So that's kind of an thing to see there.
Sorry, Colin, those were all greater than 90 centimeters.
These are all greater than 90 centimeter females. Yeah, we specifically wanted to target a little bit bigger females to get hopefully later developing females that we can use to help build our audiometric curve. Yeah. And then last thing, so we've tried to histologically stage all of our fecundity samples to be able to weed out samples that we're not going to be able to use for fecundity estimation, because you have to have females that are developed far enough along in order to be able to do a fecundity estimation. If it's too early in the process, you might not be able to separate the oocytes from the tissue because they're not developed far enough. And so we've been able to stage all of our 2023, 2024 FIS samples, as well as all the special collections in 24 and 25 in region 2. And what we did was we just, I created some quick GSI plots comparing GSI or gonatosomatic index to age, weight, and length. And what you see here is the 4 different plots are the 4 different stages of development. And what we wanted to see here is if there was any early indications of what you would think to be, if you think that a specific female is indicative of hyperallometry, you would expect that oocyte size is going to get proportionally way bigger than the total body mass, right? Because of egg output. So our previous studies on histology back in 2017 and 2018 looked at oocyte diameters through developmental stages. And we found that there's a definite endpoint to the size of the egg for Pacific halibut. It doesn't, like, as the gonad keeps getting bigger, the eggs don't just keep getting bigger, right? So you would expect that if a female is going to produce more eggs, that ovary size needs to get bigger, bigger, bigger. Otherwise, it's not going to hit that. Otherwise, you might think it could just be an isometric relationship. So here you have age, and there's not really a whole lot going on there. And then here's with weight. There's a whole bunch, especially as you get to further developed females. So on the bottom too, with VTG3 and GVM, there's a lot more variability in the size of the ovary compared to the overall size of the female. And then also here's length. So these plots can give us a little bit of an early indication that there might be an isometric relationship with fecundity and female Pacific halibut. Now, we really won't know the answer to that until we actually start looking at oocyte diameter and actual weights of the ovaries and extrapolating that out to fecundity estimations.
If I may interrupt for a
Yeah.
second, just for reference, the phylogenic one stage oocyte is 500 microns, half a millimeter. A GVM, which is germinal vesicle migration,
1500.
which is the first step in towards oocyte maturation. Those oocytes, the average is 1.2, 1.3 millimeters.
Yeah.
And then
Yeah, so on average, when you hit perioovulatory, which is a fully hydrated oocyte before expelling, it's on average about 2,000 microns. So it's about 2 millimeters in size, basically. And that's kind of like the definitive size for a oocyte that's going to be expelled for females. So this is just kind of an early look at, and then this summer, we're going to begin to actually start developing our methods in the lab and actually processing samples. And then hopefully in September, I might be able to present some initial preliminary data as we move forward with this. Because like what we talked about last year, the goal is to hopefully implement this with the next full assessment model, which is on a 3-year cycle. Last year was the last full assessment. So in a 3-year, we'll be able to hopefully populate this and see where this goes for the next assessment.
Here, remind me the assessment, spawning output is in terms of number of eggs.
It's in terms of spawning biomass right now, but algebraically it doesn't matter. So it's, if we had a fecundity relationship that was greater than proportional to biomass, it would just be multiplication. So I could assign an arbitrary eggs per gram and call it egg output now. But
That wouldn't matter, it's useful for you. But if there is a,
then we would have if we had a relationship where you actually had eggs per gram by size, we could that would definitely matter. But early indications are we're not seeing much evidence of that. But algebraically, the fecundity and maturity, it's going to be egg output at age. So the two are going to be multiplied the same way. And similarly, if we get to the point where we have the resolution in the data, it's not a problem to add to make that time variant as well. If we think we can understand the mechanism. We didn't do that with the initial look at maturity because we didn't feel like with 3 years of histology, we could necessarily delineate signal from noise yet. So average stuff like that. But that's as we develop more years of information, we may decide that time varying reproductive output
sure, it looked like it there.
measurable. Yeah. Yeah.
Also, another thing is that, currently there is only one ogive in the assessment. And we do, I didn't show any of the plots here, but, we are seeing large differences regionally on when females are maturing. And so that's definitely something to look at as far as do those trends continue through time and how that impacts stuff. Right now, we're just lumping it all into one coast-wide curve.
But as we discussed at the earlier SRB meetings, we did weight that based on the
Yeah,
estimates
yeah.
of density,
That's right.
abundance in the areas. So it is still representative
Yeah.
of the overall population, even though we have areas, some differences in areas.
Representative of one distribution, the overall population, right? So if that shifts through time, it's a
longer. Correct. So it's weighted and then averaged for what we're doing now. But if we were to go to a time varying approach, we have the weightings for the years in which we have surveys. So we could extend that out and maintain that biomass distribution, or maintain the effect of the biomass distribution with different maturity by region if we needed to.
Yeah, so the spatial distribution is to weight that, that's from.
It's from the years in which.
It's from back, yeah, so I can go.
It's from.
I'll go back real quick to go through it. It's good to,
Yeah.
Every so often, it's actually good for me to run through this to remember exactly the process. So I'll use this. So these curves here are based off of, so we created regional curves. So every year.
Annual regional.
Yeah, exactly. I don't show those plots here. So
So some of this is temporal, but some of this could also be shifts in the spatial distribution.
exactly through time. Yeah.
Have you done that separately, holding the spatial weighting fixed?
I don't think we have.
You-
No,
you
we
could
have.
Separate those out.
Yeah, that would be a good thing to look at. And that's something that we haven't done. Yeah, so right now, each one of these colored curves is 4 regional curves that are weighted and then averaged or pooled over a 3-year time window. So like 2005 is actually 4, 5, 6. And the reason why we did that, one was we had 3 years of histological data. So it matched the time series of the histology calibration. And also because of recent reductions in our set line survey, that 3-year time window allowed us to incorporate all 4 biological regions within one coastwide curve. And if we did that, you would start seeing in 2022, 2023, it wouldn't be incorporating all 4 biological regions because we type reductions in our FIS. So that's another reason why it's important that we sample across all 4 biological regions on our FIS. But, dating back to the older years, that wasn't an issue, but it's more recently that we've started running into that issue where we don't have visual maturity data across all 4 regions every year now, more recently. Yeah.
I guess I'm jumping ahead and trying to figure out whether it matters whether these shifts in the curves are because the whole population is maturing earlier, say, or more of the population is in an area where fish mature earlier. That probably doesn't.
It might.
Might matter.
Yeah. Especially when you start thinking about what are the consequences of spatial management decisions. Things like that. If you start getting fish concentrating more in some regions, that's where the majority of spawning is coming from. And it might be different than historically or something. You might end up with something that ends up being sensitive to allocation in regions or something.
So we should ask for.
That's a great point, because currently, regions 2 and region 3 have the highest abundance estimates across the coastwide stock. Region 3 in particular, we're seeing way earlier maturation compared to region 2. We are seeing a little bit later maturing fish in region 2, but in the central Gulf of Alaska is where we're seeing the youngest maturing females. We're seeing I think if you just looked at the 2024 data alone, I think in the central Gulf, we had an 850 of 8.5, which is really young.
It does get a little more complicated though, because these fish move
Yeah.
over their lifetime. So they weren't necessarily.
This is a snapshot in time, right?
Not necessarily from where we measured them. And they also move for spawning. So where we look at them in the summertime could be 1,000 kilometers from where they're spawning in the winter. So there's a lot of levels of spatial dynamics. Yeah. Which is why our initial cut here was to try and create population level estimates of maturity schedule that would be most applicable to a coastwide assessment.
One other thing, how paired are the samples you're genotyping versus doing histology? Do you have genotypes on all the individuals you're doing histology work on?
We do have fin clips for all of them. I don't think they haven't been processed, but we do have fin clips for every fish that has a histology sample.
The only ones I can think are those, 2020 winter collections and maybe in the 2018.
Yeah.
But
No, definitely.
But those weren't included in the maturity estimates.
oh, sure.
That data was not included. Yeah, that's right. But we do have every fish that gets processed on the survey with like an otolith and all that, they all get a fin clip as well. So every histology sample does have a fin clip. So that is something that can be pursued.
Yeah, so one of the things I'm just thinking, this is more, well, it's both ecology and management related in terms of how you calculate reference points, but the idea of going for eggs as a setting management reference points for how many females should be out there, you're kind of assuming that all eggs are created. And if you actually start seeing a shift from large to small or small to large eggs in the population, and those things might be genetically determined, you can actually track it. Yeah, find the markers that relate to things like egg size and stuff like that. If the environment is changing in such a way that really big eggs have a preferential survival, and you start to see that shift in your fish, there might be something interesting to look at. Might end up having the data to be able to.
Yeah, that's another one of the benefits of this autodiametric method is every sample, you're able to get full classification of oocyte diameter size for every individual. And that is something as you continue to keep doing this years and years and years, you could be able to see if there's any shifts in size of eggs. So like younger maturing females, are their eggs smaller? Are older maturing females, are their eggs bigger, right? So you're able to see some differences there. That would definitely be interesting to see. I think I just had just one last slide here. Yeah, so we are continuing to collect samples on the 2026 setline survey. They're actually having training right now in the other room. So they're hitting the water next week. The goal is to collect maturity samples across all 4 regions again. Right now, there isn't a boat contracted for 4B, but we're all hopeful that is going to happen this year. And then we're going to focus on fecundity collections in northern region 2, so in the far eastern part of the Gulf of Alaska, and then also basically all the regions that are being sampled out west. So it's kind of the central to western Gulf of Alaska, and then out in the Aleutian Islands, we'll be collecting some kind of samples again. So, and I think that's all I got.
Would this be a time for a break?
Sounds good, unless there's any immediate additional questions. If not, then let's break for, I think we're scheduled for a half-hour break, but might come back a little bit early. Not even lunch is a Tuesday anyway. Probably about 20 minutes or so, 15, 20 minutes. All right, thank you. Welcome back. Over to you, Chair.
Okay, we're going to continue on with growth.
Okay, thank you. We'll move on to the third research area. That's the area of growth. The SRB has heard in the past our previous efforts at understanding the physiological basis of growth in Pacific halibut. We conducted a few studies, laboratory studies, evaluating the effects of temperature and food ration on several physiological growth markers. This work resulted in a publication last year in 2025 in the Journal of Experimental Biology, in which we described those physiological growth markers and their possible application to inform on growth status and juvenile Pacific halibut. But in the meantime, what the Secretary has been doing is thinking about ways to incorporate this and other sources of information into a better understanding on the effects of drivers and changes in size of age. And this has become one of the most important questions for the secretariat at this point. And we have a few ideas that we'll present to you tomorrow during discussion session to help us guide future efforts and work in this area, size of age. The other area that I'd like to report is on the mortality and survival assessment. This is an area where we've conducted quite a bit of work over the last few years. Contemplated work in two particular areas. One was in estimating discard mortality rates in the longline fishery, and this study was completed and published. We actually published 3 papers on this work. And a similar project was undertaken to estimate discard mortality rates from fish caught and released from the recreational fishery. So the relevance for stock assessment was to try to improve estimates of discard mortality for both types of fisheries in order to reduce potential bias in stock assessment results and management of mortality rates. On this last project, the work is completed, and now we have a manuscript that is in preparation in which we're going to be reporting on the experimental derived mortality rates for the recreational fishery and some description of capture conditions and how those affect liabilities and mortality. So with that being said, we're going to move on to the last one, last area, which is fishing technology. And we've been focusing, as you will hear from Claude in just a second, we've been focusing on efforts to reduce whale depredation in the Pacific longline fishery. So, Claude.
Right. Thank you. So this is just a kind of a quick retouch on this project that we've been working on. This was the 3rd phase. The first phase was an international workshop with experts and affected fishers that helped identify 2 devices that we then piloted as the second phase. One was a shuttle that encloses the fish inside of it and protects them on the way to the surface. The other one was a kind of a branch line, a modified type of long line gear that had shrouds on each branch that would slide over the catch again on the way up to the surface. The pilot work showed that the shuttle was the kind of the tool that was going to have a better outcome and likelihood of full protection of that catch. And kind of the concept of how it works. So typically depredation that we see in the Pacific halibut fleet is happening during the haulback event. So the whales are in the area during the haulback event. You're bringing the fish up through the water column, exposing them to the whales who are biting them off. They're either removing them completely or they are taking chunks out of them or leaving just lips or heads behind. And so we worked with a company out of Norway called Sago Solutions, who've developed this shuttle, which is essentially a large aluminum and steel container that is threaded onto the ground line, and it slides down the gear to near the ocean floor. It has a hinged door at the front. The gear comes in that door, and so the hooked fish come inside of it. They get released inside the shuttle. The empty hooks go out the backside of the shuttle. It then encounters a pre-installed stopper, which is the device you see in the lower right-hand side there, which then is meant to close the door up and then act as a blocker that helps lift the device all the way through the water column up to the surface, protecting the catch inside of it from the whales eating them. And then once it's at the surface, the device gets hoisted up on a crane and boom system, swung onto the vessel, and then there's a hinged door at the bottom of the shuttle that is opened up to release the catch onto the vessel. So last late May, we went out and tested this device. This was the 3rd phase of the testing in the presence of depredating whales. to better kind of categorize its performance and kind of refine its attachment to the gear. So the preliminary results on this, we've completed analysis using a bunch of video cameras we had on the gear. We've got through 10 out of the 15 sets. This is looking at over about 5,000 observations. And essentially, it's good at retaining Pacific halibut. You get very few exclusions, no escapees. You get some small halibut that go right through the device and back out the other side. But overall, you get a pretty good retention rate of about 91%. Sablefish, you do not get as good a retention rates. You get a lot more escapees and pass-throughs. And we believe that can be pretty easily mitigated by paying better attention to the stopper, how it sets into the device. We had a couple cases where it did not sit properly into the mouth of the device, so it's partly unblocked, and as it's coming up, it's still lifting the device up, but it's partially unblocked, and the sablefish just drift right out of it or actively swim. But it's pretty easy to dial that in. There's also tines, like a spring-loaded door that they're coming in, and you can easily change the number of tines so that the spacing is narrower again, which would reduce sablefish escapes, which is something that's of interest to the fishery because they often have both black cod or sablefish quota in conjunction with their halibut quota. The other thing we looked at, which is of interest to the fishers, is are we losing or excluding Pacific halibut of certain sizes? They don't want to lose the big ones. And this is from over 500 observations where we have very similar sized halibut being caught within the shuttle as in compared to the unprotected gear. We had opportunity and funding to continue the work until late October. So the vessel was very interested in the use of the device. So they wanted to go out and do some commercial efforts with it. We sent a field specialist to collect biological information on the sets. They did 2 trips in October, but due to a combination of weather challenges and lack of whales, they ended up only setting the device on a total of 4 sets on that work, but we'll compile that in along with the other data that's been collected over this final set. And I didn't mention, but on the earlier work there, out of those 15 sets with both control gear and shuttle gear, there were 6 sets that did have whales actively depredating on the gear. So overall, the conclusions are that the shuttle can be safely deployed by vessels that have a picking boom. This was a fairly small vessel that we deployed it on. It was 57.5 feet or 17 meters long. The shuttle had good retention in terms of numbers and the sizes of Pacific halibut. We had up to 1,800 pounds of halibut inside the device, and that was about a third full. So it could certainly hold more. And there's certainly continued interest from the fleet in this approach. And then that's just a summary of some of the research grants here on the fishing technology side. And I guess the methylated is on here too, Giselle. So any questions on that study?
I guess there weren't that many sets with whales there, but do you have any preliminary data on how effective it is at reducing depredation? I mean, can the whales still get the halibut when you use the shuttle?
Right. So we had 6 sets that we had the whales on, but we had highly variable catch comparisons between the control and the shuttle. And some of that was because we got tangled up with ghost fishing gear. So we would be trying to haul it and we'd get stuff tangled up in some of the hooks, and so that confounded it. We had a set where we got really excited because we had a whole bunch of lips only coming up. So we knew the whales were actively feeding on it. And then when the shuttle came up, it had no halibut in it, but it had rockfish only in it. And we could tell that the gear was set all as one set with a blank space or a blank half skate of gear where we could thread the unit on without lifting fish into the water column. And we had cameras on the shuttle the whole time, both externally and internally, to monitor lost fish that are getting excluded and things like that. But it appeared that on that particular set, the set went from one type of fishing ground into a different type of fishing ground, and we started encountering different species. And so we went from kind of flatfish habitat to exclusively rockfish habitat in that set. And so it didn't feel like it was representative. There was enough there to tease the fishermen that they felt this was definitely a solution that could work, and they would like to work with it more. But I'd say it was inconclusive on our actual test results when you do the side-by-side comparisons. So that was a little frustrating, but that was the reality of what the fishing produced.
They want to work with it more, like be paid to work with it, or buy one of those things?
To be able to test it and trial it out more in their field. So
I just think those are two really different levels of interest. Right?
they are, yeah. But the fact that, even with inconclusive results, that they felt this had potential, that they wanted to go and actively use it, and that we've gotten similar questions about it, I think is an indication that they definitely see the potential in it and felt that the conditions we were fishing in didn't fully highlight its capabilities.
So what's the next stage? Are they going to test it on their own? Do you find grant money to keep testing it?
Yeah, I think right now to keep moving forward, it would probably need either one of those solutions. And right now, I think the grant money area, this has largely been funded through the bycatch reduction engineering program. And that money right now appears to be included going forward. And so I think we'd have to find another source, or the fishermen would have to find a way to fund that research. We have the 2 units available, but we're not actively pursuing people to test it right now. But certainly, I think we'd be open to discussions if some funding was available, or if they were able to do the work on their own. I think one of the concerns about it is there's a size limit in the fishery, and it certainly causes the release of the fish without doing careful release like they do at the side of the boat causes more severe injuries. So if you were to release small fish here, they'd have a higher or a poorer survivability because of the facial damage from the hook releases. So I think consideration would have to be given there to move forward with it. Either if you were going to continue testing it, you'd probably want an observer or a sampler of some sort to monitor some of that stuff. And then kind of on the next steps, you'd either want to consider regulations that would require full retention of fish when the device is being used, which probably makes sense. If you have depredating whales around, you probably don't want to be putting crumbs into the water to encourage them to hang around vessels. Or you could pursue something like a hook design or a release design inside the device that is producing less damage, very similar to a careful release. And certainly the inventor of the device has ideas on that front, but we'd have to identify and secure funding to pursue that if we wanted to do that. So I think right now we're probably looking at the end of the testing of this gear for now.
The idea with it was always that it would just be used when whales are around.
That's right. It's meant to be a tool that the fishers could use when whales are around.
So that 90% retention rate could be twice as good as what you get with whales without depredation. Right? It shouldn't be compared to 100%.
Yes,
because that's not the alternative.
That's right.
The same with the survival or release survival, right?
Mm-hmm.
Having returning fish to the water with missing their jaws could be about equivalent to returning them to the water just to get eaten.
Right. Sure.
Is there any benefit to sort of human safety from these in terms of not being as prone to getting hooking injuries on the pullback?
I don't think so. There's not a high risk there to begin with. These guys are used to using and releasing hooks, and the hooks are still coming to the surface, right? They're coming ahead of the device, so they're not having to physically remove the fish, but they're so efficient at that I don't think that's a big issue. Certainly, there's some safety issues that need to be considered on different size vessels and how high your gunnel is when you're if it's a full device and it's swinging around in marginal conditions potentially. But it's also a device that's allowing them to recover their catch when there's whales around, where other solutions that have been used, like slinky pots, are now getting, the whales have adapted to them and are now tearing them open and getting jackpotted with a whole bunch of fish at once. Whereas this device, that can't happen. So trying to find something where the fishermen can use a tool and stay on the grounds, as opposed to trying to slough the whales off to another fisher or run away from them, which is very costly in fuel, time, and wages. I think this is a potential solution. Other considerations might be to make it more modular, where you could crack the device in half and clip it back together to make it easier to stow on the vessel, because it does take up this sizable amount of space. Or something collapsible, kind of like a large dog crate, where it just snaps into position when you need it and you fold it back up when you don't need it. I think those would be desirable features. But that would probably be more something where the manufacturer would have to pursue that. We're more trying to demonstrate that the device can be used in this fleet and it can have a positive effect on the catch rates in the presence of whales. So,
are there estimates of these sort of whale effects, either the sort of frequency with which the fishers have to change sites or move, or the amount of catch that's lost directly through predation?
there are challenging estimates to come up with
sure.
for a variety of reasons, but it's been estimated that it's anywhere from for Pacific halibut, anywhere from 15 to 37% of the sets are affected in certain regions, and then the catch rate is anywhere from kind of 15 to 70% of the fish being removed. It's hard to tell sometimes if the hook was just empty and the bait had been consumed by other dead predators, whether that's crabs on the bottom or hagfish or something like that, or if whales are doing it. It seems like the killer whales leave more evidence. They're a little more sloppy in how they do their behavior. Sperm whales are much more effective at removing whole fish. But then you're like, was there ever a fish on there, or did something just take the bait off? And so because it's largely unseen behavior, it's hard to tease that out. But those are the kind of the ranges that we're seeing. And those numbers were published back in 2013, 14. So they're a bit old now. The behavior is expanding, certainly with killer whales. It's becoming much more widespread. So
I'm sure it's a dinner bell for them.
Yeah.
Regarding the sperm whale predation, are there ever efforts to put a hydrophone on the long line in order to see if they happen to be around at that time?
Yeah, so there's a group out of Southeast Alaska called Alpha, Alaska Longline Fisheries Association or something like that. And they've been doing some active work on kind of monitoring where the whales are with hydrophones. So they'll have like a hose behind the boat that they drag that has several hydrophones in it and allows them to triangulate on where the whales are. And they are trying to use that to kind of have some spatial and temporal avoidance strategies within the fleet. And they've had some limited success with that. The idea of using hydrophones to understand how many are being depredated has been attempted, but again, it's without knowledge of exactly how many fish were on the bottom, it's hard to tell, but they certainly can tell that the animals are eating, because you get these creak sounds when they're actively targeting their fish. And so there have been some studies that have incorporated that. We have not tested this device in the presence of sperm whales, so we didn't pursue getting hydrophones for that work. But there are collaborators interested in that. But again, I think for further work, we'd have to find a source. Seems for this work anyways, the key funding sources have dried up. So.
Great, thanks.
Well, if there are no further questions, this is basically the end of the biological system science update.
That's good. Joseph, is there anything more planned in terms of growth? You got that paper out.
Yeah, well, that's what I mentioned before, is that we are brainstorming a few ideas to conduct work on looking at the offensive drivers of Saturday, and we're going to be presenting those to you tomorrow for discussion.
Okay.
These are very preliminary ideas, so we really look forward to your input in identifying those key areas that we should be pursuing. So there's going to be a few ideas that we're going to be proposing, and then let's just have a good discussion on the.
Sounds great. Yeah, I just wanted to make sure
We move on to stock assessment.
Yeah. Just presentation 07, please.
Just make you presenter. Great. Okay. Well, I'm going to give you an update on the development of this year's stock assessment. So the components I'm going to speak to are, I'll do a little refresher on our stock assessment process. We've mentioned it a couple times, but just to kind of orient you to the schedule that we're on and the various documents that are produced and how everything fits together. I'll talk a little bit about just the routine time series and software updates that have already been done this spring. And then I'll speak to the two outstanding SRB requests that I had to work on for this particular meeting, and then give you a little prospectus on what the rest of this year looks like. So as was mentioned, I think Colin mentioned earlier, we are on a schedule where we do a full stock assessment roughly every 3 years. So historically, these have been in 2015, 19, 22, and 2025, last year being the most recent full stock assessment. When we do a full stock assessment, that's a full reevaluation. So we basically take all the data sets down to the studs, rebuild them, any updates that have to happen, changes in analysis, pre-processing, anything like that. And then we do the same with the stock assessment models as well. I'll speak to what models we're using in another slide here. So that process occurred last year, and perhaps just to orient you to the documentation that we produce as part of that. So it was mentioned earlier that the sensitivities to the maturity and other things were provided as part of the June SRB meeting documents for last year. And that's the general schedule. So in a year with a full assessment, there'll be a big document produced in June that describes, it has all the technical details and model diagnostics. We'll provide the model files and output and all that sort of thing for the June meeting in a full stock assessment year. Then there'll be a follow-up in the fall to all the questions and suggestions and changes that arose. We'll clean that up, and then we'll move on to the rest of the assessment. On our website, we have a page for the stock assessment. And you can actually go back and see all the stock assessments back to the late 1970s, back when we were using equilibrium models as a stock assessment. So they're all there. Every year stock assessment is there. Since about 2012, there's more detail provided there. In the annual schedule, and I'm going to take a minute just to describe it, because we produced that document for the SRB. And then this year, you see it's a fairly abbreviated document, mainly focusing on requests for this meeting. In the fall, we will produce what is essentially an executive summary for the commission's use at the interim meeting. And then again, at the annual meeting, we'll have effectively an executive summary. It's 10 or 15 pages long, provides the salient results from the stock assessment. We also provide a parallel document, which provides the immediate term projections, which we refer to as the next 3 years, for decision-making purposes. By request of the commission, we no longer provide the large stock assessment documents as part of the meeting materials. What we do every year, publish a summary of data sources, which would normally be a chapter or a section in a larger stock assessment document. We separate it into two documents. We have a data summary, and then we have a longer stock assessment document. Those get published directly to our website. So that's what you'll find on the website. And those go back every year. So there's a lot of different documents that are produced every year associated with the stock assessment. But you'll find at the end of the day, you'll find those all on the stock assessment webpage. So this year, getting back to where we are now, this year is an update So it's the first year in between full stock assessments. As Colin mentioned, we're already targeting the fecundity analysis as one of the major avenues of investigation for 2028 full stock assessment. And one of these requests here with regard to state space models will be probably the other. So I'll speak to that. But for this year, we generally only make minor, or if we come across something we have to change, necessary changes to data sets and models. You know, generally we try to just add the new information.
Ian, just because I was thinking about it when I was reading through all the stuff, where would I be able to find like the full technical description, like all the equations that describe the assessment assumptions that are being used?
The June document for the on years would be the broadest technical description. I haven't recreated all of the equations because all 4 models are implemented in stock synthesis, and that's published, and so all those equations are there. The input files, including the control file, which shows what all of the various features that are engaged in each of the models, is provided on the website. And so somebody could go through and cross-check and see exactly what features are turned on, what settings are. Much of that is summarized in the document, primary leading parameter estimates, uncertainty, that sort of thing. A summary of residual diagnostics and other things is also in that document. So it's the June SRB meeting document. We also, as part of that, provide, I think directly to the SRB, we provide the output files to look at the really detailed diagnostics, but we don't publish them. But we provide that in those June meetings. It's made a little bit more challenging because we're using an ensemble of 4 models, which is what I'm going to speak to on the next slide. So we, providing the same level of individual data point diagnostics that you might get in a single stock assessment, a single model stock assessment is a little bit more challenging. And that's why we go to the packaged format for the SRB meeting in June. We went to this format of producing the executive summary for the big meetings because essentially the commission process said too much information. We don't need to see all the diagnostics. We don't need to see all the summary of the data. Just give us the executive summary, publish the rest to the website. And I recognize that most people probably never go read it. But nonetheless, we produce it every year and it is there to digest if they need to. It also contains some tables and figures that don't make it into the executive summary, so there's a better record of that information.
One thing I noted about that, the input files are in PDF format.
Yes. Yep.
That seems like kind of neither fish nor fowl. Like you can't use them to.
You would have to do a little bit of organizing. Yeah, the idea there is not so much that somebody can go run it, but that they can go look and see it. And generally, the thinking behind that, and we provide, for example, the input data is there as well. And we provide that out if somebody asks for it, provide it. The benefit of having it as a PDF and having people ask for a native file is that we can generally have a sense of who's using it. And have the opportunity to ask them, hey, if you're doing a research project on this particular, is it at least close to the right? The flip side being, if you just post the file, then but we're not. We don't restrict access. People ask for them. So this is a bit of a long-winded introduction, but I thought, Mike, at least since you're sort of new to the process and we have 100 years of history behind virtually everything we do, it might not hurt to take a few extra minutes to appreciate why. So since 2013, we have used an ensemble of models for our stock assessment. So it's essentially we're model averaging 4 different models. These models represent a cross of 2 different approaches to treating the information. We have 2 models, our long time series starting in the late 1800s with a full time series of catch history. We have catch at age information going back to 1935. It's a pretty incredible data set to work with. CPUE going all the way back to 1907, I think. And that's crossed with two different ways to treat the data. So two of the models are coastwide models. Everything's aggregated up to the fishery and the survey. And the other two models are disaggregated to biological regions. So we do an areas as fleets approach where we have essentially 4 fisheries. We have the fishery in region 2, region 3, region 4, and 4B, and we allow for some of the differences in those fisheries to come through via sector selectivity curves. Not an actual spatial model, just a proxy for space. And so each year, these 4 models produce different results. This is actually the summary from last year's stock assessment of the results for the beginning of year 2026 spawning biomass. And you can see that they're all in the ballpark, but not full agreement. And we integrate those on the bottom panel there into essentially a single set of results for use. The banding indicates the relative uncertainty, and that's again integrated across all 4 models. We do that using the maximum likelihood estimates and the asymptotic estimates of uncertainty, and essentially just do a parametric bootstrap out of that. We've spent years discussing weighting of these models. We've looked at retrospective performance. We've looked at predictive performance. We've looked at model diagnostics of all types. And although we've, in certain years, certain methods look better than others, we've never broken from just equally weighting. They all seem like, and the bar for getting into the ensemble is that the model has to be internally consistent, and it has to represent a coherent enough and logically consistent hypothesis that if that was the only model on the table, we would probably use it. We don't try to do what some places do, which is to create a huge ensemble of all the various combinations of parameters and things, including some things that might be completely implausible on their own. We've taken a different road of just having these 4 models.
Who does that?
Tuna. There's a couple tuna assessments where they do these like super ensembles, 1,200 models, and they.
Anything Lori Kel's involved.
Yeah, and there's some benefit in that because they're essentially integrating over that space with brute force. But the downside is they often equally weight all those, and some of those combinations of like really low natural mortality and really low steepness, and some of those combinations are basically impossible. Yeah, and so they have these resampling algorithms to try and exclude some of the things where the population's extinct, doesn't fit the data. But I think there's a lot less thought given to fitting the data in the first place, especially for a lot of the out combinations. So it's not, there's different ways to go about it. The way we've gone about it is to say, look, these are, if you took 4 seasoned stock assessment scientists and put them together or separated them, didn't put them together, they could each come up with one of these 4 models and probably sell it to a review and say, this is a pretty decent model for halibut. Even within Seattle, there are camps of, one of the science centers tends to do long time series models and the other tends to do short time series models. Our part of our thinking here is we want to be a little more robust to structural uncertainty. And this is one way to do it. What we've found, and actually Alan and I published a paper about 6 or 7 years ago, we've found that these models respond differently to new data. So we have, they have some consistency, but in some years, the new data points will pull a couple models up and push a couple down and vice versa. And it's not always the same models. And by the central limit theorem, having several models tends to make the whole process a little bit more stable. Which, if you don't know what the true, if you don't know what the true answer is, stability is not a bad thing. And over the, I guess now 13 years we've been using an ensemble, we've yet to come up with a reason to chuck any of these models out. However, as you'll see, we got some ideas about adding some new models to the mix as well.
Ian, you said that each of these represents something that assessment scientists might come up with. I was talking with Pat Sullivan last week because he was at a meeting that Mike and I were both at, and talking about how as the assessment scientist has changed through history, the approaches have changed also. And I realized I've never seen a comparison across the different eras of assessment here to see, sort of what has changed and whether there are any models from the past that might be worth resurrecting to put in this ensemble, or whether there's been kind of an evolution just getting better and better. So we wouldn't want to go back.
So Pat hates ensembles. That's a fact. And we actually, Steve Martell and I published a paper in 2015, which provided a history of the stock assessment modeling for the IPHC. And it's a great cast of characters. Terry Quinn and Rick DeRiso are in there. Pat's in there. Anna Parma's in there. There's a lot of big names that have done stock assessments. And we've done everything from equilibrium models to, we had in the 80s, we had KGN, which was a catch at age using tagging data, fully spatially explicit. And mapped on top of all of that, we've also had some horrific retrospective patterns. We've had at least 4 runs of really bad retrospective patterns going in both directions, some of the good kind and some of the bad kind. And so my short answer would be no, there's none of those models that are probably something we want to bring back.
You went beyond them for good reason.
Yeah, I think we have moved beyond them. Maybe let's talk a little bit more about that when we get to the state space discussion. But it's, yeah. Okay, so that's just a little bit of history to preface the next parts of the discussion. So the routine things that I do every year, the first thing that I do after the annual meeting occurs, so produce the assessment, which produces spawning biomass estimates as of the beginning of the year and a 3-year projection based on different catch levels, and that's what the decision making is based on. The first thing I do after the dust settles from the meeting is to extend all the models by 1 year and to include the mortality that we've projected based on the adopted catch levels, because that provides the basis for all the rest of the comparisons. Then we can see, as we step forward, I always provide a bridging analysis. We can see, okay, this is what we thought the mortality was. This is what it actually turned out to be, and this is how much difference that made. So there's that step in there. And then we can also see in a stepwise manner all of the various changes. And of course, I don't always produce a bridge that has every link in the chain, because most of them aren't very interesting. But what we do is we add each data set as they come in the fall, and then we identify the ones that had the biggest change and highlight 2 or 3 of those, if they're there, that had the biggest effect on the year's stock assessment. So we do that every year to provide a bridge so that people can understand, if the assessment results change, what was driving that. Was it the fact that we projected the wrong mortality? Was it the fact that we made a change somewhere in the assessment? Was it the fact that we got new data that told us something different? And all of those over the last 15 years have been the case at least once or twice. I don't have any of that to show you now because we haven't done anything that's interesting. None of this actually makes any difference, but I'm just setting the stage for what you'll see, what the commission will see down the road. In some years, there could be a software update. In early years of stock synthesis, there were bugs all the time, and they made a difference sometimes. We haven't had any changes to stock synthesis in the last, I would say, 10 years that really had any effect. And again, this year, we stay up to date with the most recent version because they do find bugs and things do change. But again, this year there was no change, so that didn't have any effect on any of the output from any of these. I always try to do this step though early in the year in case something does change. It gives us the rest of the year to figure it out. Because of our compressed schedule, the last thing I want to do is get to the beginning of November when I have like 9 working days to complete the assessment and have something change in the software. That's not okay. Okay, so that's kind of where we're at in this year's process. And I didn't produce any graphics because nothing changed. There's nothing to show. The first request is actually a holdover from back in SRB026. So that would have been last June. And this is one that, again, we'll be working on over the next couple of years. And this is really our collective acknowledgement that state-space modeling is taking over everybody's stock assessments slowly, and we're going to need to be there sooner rather than later. So I think the goal for the 2028 stock assessment is to have at least a candidate or 2 state space model to either add to the ensemble or replace one of the models in there, or at least be on the table for discussion. So this is the first stages of that. Yeah, so halibut, of course, is a complicated stock assessment, and there is currently nothing off the shelf that will do everything we need. And we've had this discussion quite a bit in the past about the pros and cons of writing your own versus using a platform. I think we've got that coming up, so we can talk a little bit more about that. But some of the things that we're doing in the stock assessment, in the halibut stock assessment, like time varying processes, recruitment, selectivity, catchability, those obviously lend themselves to using the state space approach. It seems quite logical that could be a big benefit for us there. We also have to have sex-specific dynamics, which the original state space models, SAM and WHAM, don't seem to have any interest in adding sex-specific dynamics to their model platforms. We also need multiple aging error matrices. As was mentioned earlier, historically, we have surface ages, which we have an imprecision and bias estimate for. We then switched to break and bake ages in 2002, although we went back and re-aged a few things earlier than that. And as you're going to hear more about, we may soon be integrating AI aging, which will have a different aging error matrix or even multiple matrices to deal with. So going to need some flexibility there. We use environmental covariates to the stock recruit function. We use the Pacific decadal oscillation as a covariate to the stock recruit function in the 2 long time series models. And so we're going to need that kind of functionality. And we also have a prior distribution on natural mortality. So each one of these is maybe not a deal breaker, but there's a bunch of things, and none of these exist in all of the various programs. So we've looked around, and this is not all of the state space models that are in use around the world, but it's the big generalized ones, including FIMS, which is still kind of on the horizon. That's the NOAA effort to create the model to end all models, which may or may not ever see.
Start.
We just did a review.
Yeah. We just reviewed it.
Oh, yeah.
3-4 weeks ago. My time's all together, but yeah.
And they are 10 years into that process and still haven't done an actual assessment in that model?
Not one used for management, but they're going to have some done by the end of this year.
They've been saying that for a while. I hope they do.
Yeah, this was the part to actually get it kicked over. So you're right, it doesn't have a lot of the functionality that Synthesis currently has, but there's a lot of good things about it in terms of the directions it's going into.
So I'll provide a little bit for the features that we're interested in, at least initially. I just did a little comparison. There's actually some of this is provided on the FIMS website. There's some comparison among model platforms, and some of it we had to piece together from other places. We have had a rolling discussion here over, and it even predates me, probably the last 30 years, about whether the commission should have a halibut specific model, and we did for a long time, or whether we should use a more general platform. We had some trouble with our model before I got here. We had some technical issues, and it was essentially because we had an ADMB based custom model that became unwieldy and difficult to update and maintain. And when I got here, we moved to using, not immediately, but shortly moved to using stock synthesis, and then adding in an ensemble of models. And so over the years, we've had a bunch of discussion about really what direction we should take in the long term. We had an independent peer review from Kevin Stokes in 2019, and this topic came up. I know Sean liked to discuss this. Sean was maybe a stronger proponent for a build your own model. It's all a trade-off. So the benefits of using a generalized platform are that it's easier to transfer among authors. I mean, there's a lot of stock synthesis users out there, and any one of them could come in and take the files that we've got and run them on tomorrow. It's much easier in terms of reviewing a model that's well documented. the R4SS package. You can produce all the diagnostics in seconds and see everything about the model. So at this point, I think our current proposal is that we would like to stick with a more generalized platform. But it's hard to say with the development of AI assisted coding, which we are enjoying a lot for all of our coding use now. It's certainly conceivable that we could even start with a generalized platform and add features. As the trick with that being, it becomes very difficult to know what your models are really doing if you start branching out and not simulation testing the interactions between all of the various features. And that's another benefit of generalized. It is. So I would say that the goal for the 2028 stock assessment and what we're starting on right now, I don't want to get to that slide yet. What we're starting with right now is we're going to build basically a toy halibut model in at least 2 or 3 of these platforms and see which maybe we can get away without some of these features, and maybe we can add a few simple features just to get it closer. And basically just get some understanding about the capability of things. All of these platforms are developing really rapidly, as you mentioned, Mike. FIMS is maybe on the cusp of actually starting to get used. Similarly, Spork and Seattle are starting to get used. I think Spork is anticipated to be used for the Saber Fish assessment this fall. Seattle was used for 1 production assessment already, and I think they've already converted about 8 other models. WAM is, of course, you guys know all about WAM being on the East Coast, being used for a lot of different.
The Seattle multi-species, or am I thinking of?
It is, but it can be turned off. The multi-species stuff can be turned off, and then it just turns into a TMB-based, states-based model. SAM, I've had quite a few discussions with Anders, and they are not going to add any sex-specific dynamics. And for halibut, we can't do without sex-specific dynamics. That really is a deal breaker. There may be, after talking to Tim, there may be some potential to reverse engineer males and females using the spatial aspects of WAM because it's fairly generalized. It's not clear that there's a lot of support for anybody actually doing that. And there was at least 1 postdoc who worked on moving in that direction, and none of it got put back into WAM. So it's not clear that that's going to be a viable platform. But so what we've done is sort of case the landscape, see what's out there. I think we have, with Spork and Seattle, we have a couple of models that are worth exploring and building at least a toy halibut model, and then starting simple and then adding features to see where we get to, and maybe FIMS will come on.
Yeah, I definitely encourage you to think of FIMS in the same framework.
They have, unfortunately, right now they have, as you can see, they have basically none of the features we need. Yeah, but it is a big effort, and I know Noah's putting a lot of resources into that effort. So we're kind of in the Coliseum here with regard to these models right now. I feel like we're where we were 25 years ago when ADMB was just kind of widely available. TMB is now widely available. So random effects are easy. And it's not clear which of these platforms is going to run the course, which one's going to be Russell Crowe 5 years from now. Things are really changing quickly. But as I've mentioned, I think that we can probably find a path through this, build some small models, and then decide whether or not we either convince the developers to add some features we need, add them ourselves, or just use, these are all going to be, well, I guess they're all, at this point, they're all open source things. We could just branch off, take that and branch off. But I think the next step is going to be to develop some working examples with a modest number of features and see where we go from there.
So I think that those will be based on simulated data, or fit them to the real.
I think we need to start with, all of these platforms have had some level of simulation testing. I think the first step will be to actually populate them with halibut data and see if we can get in the ballpark of our existing ones. And then
Yeah.
move from there. And
I mean,
then,
the downside, of course, is you're always comparing to the existing models, so learning something. really
Well,
new is a proposition.
could be exciting. Yeah, we might learn something new. I would say if we start adding features on our own, we'll certainly need to do some simulation testing to make sure that they're.
Yeah, I would be thinking of it in terms of, there's some of the models that you do that would be easier to port into it, like just one member of the ensemble or something like that. Think of it in that framework. It's like the long time series, everything just all together would be the easiest one, it would seem to me, to try to start with the port.
Yeah, we're actually starting with the short time series. So I've actually got a Seattle model. So one of the issues is that I haven't seen a state space implementation that has 30 ages and 100 years of information and 2 sexes.
Mhm.
So just the random effects for numbers at age and F and six fleets in the model. You know, that's a lot bigger than anything that I've seen built. So the plan was to start with the short time series model with just a couple of fleets, just get the halibut data in there and start to get it in the ballpark, see where it goes. I don't think that will limit us. I mean, we may find that some of these, we get some different estimates out of these models. And so our goal isn't to limit it to things that look like the current halibut model, but I think it is going to be important to see which data sets we need to, we can get in there, and which assumptions and features are going to be most important to replicate a reasonable halibut.
Which ones will even work when you start trying to turn them on to is an interesting thing. We tried to develop a state space blue crab model that it's a monthly time step, but it has 360 time steps as part of it because we have 30 years and 3 sexes. And none of the state space stuff works with the data we have, but it's also length based. It's not age based, so it's not as straightforward to estimate everything. But and it takes about 15 to 20 minutes to do one run of it, even without the state space stuff turned on. And so the information when you have the state space stuff all turned on that you actually need to estimate everything, and when you have multiple things varying over time that you're trying to model as random effects, it becomes tricky. I reverted back to just using penalized likelihood random ones for a lot of things because the estimation of the variance terms for the random effects was not playing nicely. So especially once you start turning multiple things on, it's really unclear what's going to happen sometimes.
Yeah, so I think that's exactly what we don't want to get to right away. I think we want to start pretty simple and pretty small, get core halibut features in there, and then see. But we got a couple of years to do it. So, and we've got, like I said, I've already got a Seattle model working with a little bit of halibut data in it. Just in a single species context, no species interactions. The other features that are out there in a state space framework, some of the dynamic structural equation modeling to incorporate environmental covariances, which is pretty cool. The work that Jim Thorson and Bo Monahan and others have been doing. There's a lot of exciting stuff that this will open to. So I think this and the fecundity are probably going to be the 2 primary development aspects toward 2028.
Sounds like plenty.
Yeah, it's going to be. We're going to be busy getting this all done. Yeah, which is. We had a lunch break in there. I don't know.
12:30.
I can continue on. I really have one section left in the presentation, and it's going to take a while. So we have the option. We could break now, or I could just go through. It'd probably be maybe 30, 40 minutes, depending on how much discussion we have.
So let's keep going.
Okay, great. So the second one, this is take two on evaluating our decision table, basically. So every year we produce a decision table, and I think there was actually the decision tables in the document. I didn't put it back in the presentation, but we produce a decision table which makes a projection of the probability of the stock declining over the next 1 to 3 years, and that's one of the primary inputs for the management decisions that are made. Now I don't recall which SRB meeting it was. I guess it might have been last June, and then we redid it a little bit for SRB27, which was last fall. But I had made an attempt to try and evaluate the question that we get frequently is how well does the decision table perform? Basically, are the probabilities that you're giving us of any value, or is it just? And so that's basically we're trying to answer that question. And the SRB recommended that I look into receiver operating characteristics. And that's something I hadn't done. And it turns out these are pretty applicable to this kind of a question with a couple differences that I'll highlight as we go through. Because these are really, this approach is really designed for evaluating the efficacy of some sort of a testing procedure that it's developed around how well does your test do in detecting something. And that's pretty close to what we're doing in the decision table. So I actually think it worked pretty well. So this is the information that we have to bring to the problem. We started using the ensemble in 2013, and that's the year we started using a decision table as well. And so we have all the way back to 2013, we have on the left-hand side, this is based on the most recent stock assessment. This is what we actually think happened to the spawning biomass in each of these years. And so the percent change is the percent change from that year to the next year. And then we also have what we projected at the time with the assessment at the time. So this is not a look back from now. That's what was in the decision table at that particular time. So the probability that the spawning biomass would go down in any of those years, and you can see that there some years there was a high probability of the stock going down and it didn't, and other years there was a high probability of it going down and it did, and it went down by different levels in this whole time. I would note that it never went down by more than 9.6%. So we haven't had any major changes in a single year. And I think that has some implications for the power of our testing here. And as you'll see, I looked at a couple different thresholds to get at this, because honestly, a change of 1%, given the uncertainties in all of our data, like I actually wouldn't expect us to be able to detect changes on that range. So this is the data, in quotes, that we're putting into the analysis for the receiver operating curves. And of course, we don't know the percent change for 2026 yet, because we won't know that till next year's stock assessment. But I do include the projection that we made, because we've already made it. That's a done deal.
So you don't turn these into a binary before?
Well, so we could say that if it's at least a 50% chance of stock decline, we're saying it will decline.
No, the other way around. Whether it goes up or down in a binary. So instead of percent change, just one of the signs positive.
Yes, so I'll show you that.
Okay,
as well. That result is in there.
Yeah.
Yeah. But I wanted to show you the actual change.
That would
as
be
well.
the standard way.
Yes, and that's the first one we did, and then I'll show you a couple of others as well. So just to refresh, because this was all new to us as we put this together, the receiver operating characteristic, you're generally using this to compare and evaluate testing, or it's actually like disease detection methods. And what you're interested in is the true positive rate versus the false positive rate. In most cases, the false, let's see. In most cases, the focus is on a positive test. It's basically how often do you get your positive test right versus wrong. And that's what this approach is really around. So what we did was we compared the frequency of projecting the spawning biomass correctly going up or down with different levels of uncertainty. So essentially, we're treating the percentage in the decision table as a different test. So we might have, if we say there's a 5% chance of decline, that's one test. 10% chance of decline, that's another test. 15 would be another test. So we can classify that as different tests from the decision table. And the information that goes into the receiver operating characteristics is really just this frequency matrix of did it actually go down or not, and did you project it to go down? So like you said, there's no percentages here, and I'll talk to how we got to the percentages. But basically, there's a frequency of occurrence in each of those four quadrants: the P's and the N's. The P's are the positives projections, and the N's are the negative projections, and they're either true or false. So there's two axes of the receiver operating characteristic to create the area under the curve. The first one's the proportion of true positive observations, and the second one is the proportion of false positive observations. So this would be years where we predicted or projected the stock would decline and it actually did, would be a true positive, or we projected the stock would decline and it didn't, and that would be a false positive. So then, as I mentioned, what we did was we considered each incremental value of probability of decline in the decision table as a different test. So we can take a threshold and say, did it pass the test or not? If we predicted a 47% decline, well, it would have passed all the tests less than that and not any greater than that. So we're kind of bending the method a little bit to make it fit the decision table. It seemed like a way to go. And then we applied this to four different ways. We did this what would be the standard, which would be any decline. But then we also wanted to look, well, what if we only looked at declines of at least 1% or at least 2% or at least 3%? We could have gone farther than that, but then we run out of data. We only have 13 data. So get a little carried away here. But you might imagine that people would be obviously more interested in how do we do if the stock's going to go down by at least 3% than any decline at all, because at some point it's just. So to orient you, this is, I believe, the standard approach to plotting and evaluating this information. This is the area under the curve plot. And honestly, to be completely honest, I don't understand why they call it the area under the curve, because you're not actually integrating anything. There's nothing you can add up on these plots you're not truly comparing the area under the curve. What you're really comparing is where you land on. But the diagonal line would be a random test performance. So you'd essentially be getting as the frequency of true positives would be equal to the frequency of false positives. Say that would be random. If the actual probabilities of those things were different, you might not be exactly that line. But that's if those two things occurred in equal proportion in reality, a random performance of your test would fall on the diagonal. A perfect test would be up there in the left-hand corner. So you would get all of, when it was really positive, you would get them all right, and you would never predict a positive outcome if it didn't. So that would occur in that upper left-hand corner. So let's put our real information on here. So each of these lines connects incremental values of probability of projected decline. And we did from 5 to 95% by 5% chunks. Obviously, it's chunky because we don't have 1,000 data, so it's not a smooth curve. So the step functions are where actual data occurs. You still have the random curve on there. The red line would be any decline. So you would, if there was any decline in the stock, this is all of the various cutoffs in the decision table from 5 to 95%. See that we're doing better than a random test for predicting any kind of a decline. And as you go to a 1%, 2% in blue, and then 3% in purple, we're actually doing better. So the decision table is actually predicting when there's a bigger decline, it's got a better chance of identifying that, which is kind of what we will have happen. If we look here, I've put the points on which correspond to the median, because that's kind of the obvious breakpoint. If we say there's a 50%, at least a 50% chance of decline, that's where those points lined up. So you can see, for example, with this purple point, or let's take the blue point here. If the true decline is at least 2%, we got all those right at the median. So if there was a 50% chance of decline, we got that right if it was going to be at least 2%, and we had something in the range of 30%. We predicted about 30% of the time we predicted that it was going to decline, and it didn't decline by at least 2%. The best test, if you're just looking at, I guess, what you would call the area under the curve, is there. And that would be a 60% probability of decline as your test. So a little bit greater than the median. And in that case, for at least a 3% chance of, or at least a 3% decline in spawning biomass, we predicted those correctly 100% of the time and had a false positive rate of 25%. So I think, just cutting to the conclusions, that the decision table is actually kind of working the way we think it should. If we accept that 3% decline is important, which I think people generally would, then if the decision table has a high probability, at least 50%, maybe 60%, then I think the commission can kind of feel reassured that the stock's probably going to decline. They should probably take that information and do something with it. We're wrong in that projection about 25% of the time. So I think we can answer the question. The decision table does seem to have some predictive value. It's not perfect, but we only have 13 data points. And to be fair, the models have changed, the information's changed underneath here. So we're comparing back over this whole time period that we can. I don't know whether the models are performing better now than they were back then. We don't have enough information to get at that question, I don't think. But it's not a perfect comparison, and it's not a perfect application of receiver operating characteristics. But I think it's indicative of what we would like to see, which is that the decision table is providing some management process. So that rounds out that request. Honestly, it was kind of fun to do this analysis. It was the first way. I tried to do it graphically last time, Mike, for you, and I made some nice green and red color coded things, and yeah, maybe it was all right, but it wasn't really quantitative. So I think this is.
Yeah, when I saw the purple line.
Yeah,
this is doing what it's
somewhat
supposed to do, because I don't care a whole lot about changes less than 3%.
Exactly. And we might not even be getting those right now.
Exactly.
I mean,
Right,
that's
right.
kind of it. If we had a case, and again, we only have a short time series, if we had a case where we had a really big change in stock, obviously we would get that right. We'd learn a lot about our models overnight if we had a 25% change up or down in stuff. We haven't had anything like that.
Well, you do have several pretty big changes in there, though. You have like that series of 3 years that have pretty big negative changes.
And we did predict those. If we go back, and that's one of the things where I think actually the graphical stuff that I did have some value, because you could really see there's a bit of a lag. The stock starts to go down a little bit, and we don't pick it up necessarily in the first year, but then right away, we've got 93, 84, 95%. We've got very high probability of decline corresponding to the years when we really did have it. Yeah, it's like we are getting the signal. We don't get it instantly. Maybe we're a year behind catching up, but we generally do get the message. It'll be interesting to see this year, and I thought it was interesting to put it on here. This is the lowest probability of decline that we've projected yet. So hopefully the stock's going to actually go up, because we're saying there's really not a lot of chance of further stock based on the test they adopt.
And the other thing that we'll sometimes look at with this is P-FAIR, to make sure that that's close to 50%. So that you can actually, so the receiver operating characteristic curves can look quite good, even if your probabilities are wrong, as long as there's an increase in that event happening as your probability. So the actual probabilities can be.
Yeah, so that I think
P-FAIR is the threshold that would give you that equal chance. Yeah.
And I think that's sort of here, which we're saying, if you really want to be sure, go look at when the decision table says 60% above, you have greater confidence. That's the best performing. So if we were to create a binary, rather than reporting probabilities in the decision table, we just said, yes, it's going to go up, or no, it's going to go down. Naturally, I think we would break that at 50% to make that binary call.
And PFAIR tells you whether that's the right call or not.
Right. But in this case, I think we've backed that out by
Backed
saying
out
that
PFAIR.
60%
Yeah.
would be the best performing break point.
So that's P-opt.
Yeah.
Is 60%.
Yeah.
P-opt is the best performing.
Yeah.
PFAIR is chance.
Okay. Yeah. Okay, so let's see our operating characteristics. It was kind of a fun, well, thanks for the guidance on that one. That was, I was unaware of this type of analysis until we tried it out.
One of the other things we've tried with some of the mid-Atlantic stocks is just looking at trying to do a root mean square error for forecast predictive performance. And have you ever?
Yeah, so we did, a few years ago now, we went down quite a long rabbit hole looking at the MACE statistic, which is the predictive performance, where you basically look at your predictive skill relative to just using last year as your prediction for next year, the naive prediction. And I was actually really hoping it was going to turn out to be a great way to do model weighting, because that's what we really care about, is how well do we predict next year. And it turned out that it's messier than we thought. There's a lot of things that can affect your predictive skill. And in fact, if you have a stock like we have had for a number of these years that's not changing much, it's really difficult to beat just saying, well, last year's spawning biomass is our best prediction of next year. It doesn't mean your models are bad. It just means you don't have any contrast in your prediction. So we spent quite a lot of time on predictive performance, and ultimately just didn't turn out to be quite as useful. There's a student at the University of Washington who's working on looking at some simulation testing of the MACE statistic. Mikko and Mikko Kineen. I'm on his committee, and I definitely been encouraging this project, but he's finding that MACE isn't all that great as a model diagnostic. It's not identifying the misspecified models nearly as well as we.
That's only 2 years ago.
I know, right?
But
But there's several publications on it. I really thought it was going to be, because conceptually, really all we want out of an assessment is to be able to predict what's going to happen next year, right? I mean, that's, but
We tried a bunch of stuff over the years in terms of trying to be able to quantify predictive accuracy, and I haven't been able to find anything. So I keep hoping for you.
frustrating. I know, it's really frustrating.
To figure it out, but I started off early, part of my PhD, I was doing some stuff on AIC and BIC and DIC and looking at if any of those could be used. They didn't. We tried retrospective patterns and they weren't very useful. None of these things were better than chance or consistently worked across different scenarios when we tried to simulation test them. So I've kind of given up on it as that it's something we can actually draw from the data. The way in which we can misspecify a stock assessment model is too complicated and multifold to be able to summarize in one statistic, I think.
Right, and we never know the true answer. It's not like a storm path where we can say, exactly where it actually went after the fact. And the only times we ever know that for stock assessments when something goes horribly wrong.
Well, I haven't even been able to find one that works even when you do know the answer in simulation world. They still don't even work then. And so it's to have them in the real world is great.
Okay, so we're getting close to lunch. That rounds out that section. I wanted to just refresh you on the rest of the process from here. So we'll have the September SRB meeting. This is where we do follow up to any June requests. This being an update year, we maybe have less than we normally would in an on year. After the September meeting, the key aspects are that after the September meeting, we don't make any additional model changes unless there's something that the SRB recommends. Just change this and move forward with this change. Ideally, our two-part process allows us to show you the research in June, make the changes over the summer, see how they turned out in the fall, and then make the final decision. because our process is really compressed. The final data sets are available basically at the end of October. We close the data sets, and then by late November, we're already presenting the results to the commission. So we really don't want to have a situation where we say, hey, try this change, and then the wheels fall off the bus, and then when you put the new data in it, so we really, and we've been pretty successful in it. We do our research in the spring, we do our refinements in the summer, and then we do our production in the fall and production, so we don't make big changes.
That is a crazy time. Okay, now the other.
But this review process works really well because it as soon as the annual meeting is done, Alan and I and everybody here, we all sit down and sort of go back to the books and start doing all the research. We have, unlike the STAR panel process on the West Coast or the CDARs or all these other review processes, we're not putting our research stuff with our productions. We're not trying to finish the assessment and evaluate the research. Trying to separate. And it's worked out well in full stock assessment years because we can try out a lot of stuff. And some years, we come in with something in June and the SRB says, yeah, that was fun, but don't do that. Like some of the mace bleeding. Like we took that all the way through to here's how we could make this a production product. And, when we went back and looked and he said, look at a few more things. And then by September, we decided, well, might not really be ready. So I think we've settled on a pretty good review process in terms of just compared to the ones that I've been involved with, where often you end up at the last minute making a model change that's too big to be very comfortable when you're actually trying to do your production.
This timeline also allows us to use the current year data in the stock assessment.
Right.
That's huge.
It's something that, for better or worse, the halibut process wants an assessment that has all the recent data. We would never be able to get away. Like in some places, they have, it's two-year-old data. We would never.
I think it's a really good part of the process.
It responds quickly to new information. Sure.
I think it's what we should be trying for. That staleness.
It's not good.
It costs for it.
Yeah. So that rounds it out. The recommendations for this one is just to note the paper. And if there are things that come up as part of other discussions or this discussion, we do have the option to look at more stuff in the fall. It's always helpful for recommendations, and the SRB has been great about this in the past, to specify things that would be applicable to this fall versus things that are more long-term development. And this is a great example of these two requests. One of them is looking forward to the next full stack assessment, and the RLC stuff is for this process. And that's what I've got. So we're actually a little bit ahead of schedule because I think I had a small walk after lunch. So we're fine, even though we're 5 minutes late for lunch. We're actually still a little bit ahead.
Sounds good.
It's lunch. It surely is. What have we got? So we're supposed to come back at 1:30. I suggest a little bit later, so maybe 1:45. Okay, so we'll be back at 1:45 for those people listening. Thank you.